The first thing the camera sees is not an animal. It is a wall of black water, full of white particles moving past the lens like ash in a slow current. The ROV is already far below the last sunlight, hanging from a ship by a cable that has disappeared upward into darkness. And on the monitor in the control room, the depth number has become the most important piece of landscape. Then, the beam catches a chimney. It rises from the seafloor in broken mineral shelves, dark and rough, with a cloud of hot fluid pushing out of its mouth. For a moment it looks like smoke from a fire, except there is no fire here in the ordinary sense, and no air to carry it. There is only seawater under pressure, heated inside the crust, returning with metals, sulfide, heat, and the chemical energy that makes this place possible. Around the chimney, the seafloor is not bare. Pale crabs move over the rock. White tubes stand in clusters. Small shrimp gather where the dark fluid thins and mixes with cold water. Life is pressed close to the volcano, close enough to be fed by it, but not so close that it is destroyed. The word alien fits this scene better than it fits most things on Earth, but only if it is used carefully. These animals are not visitors from somewhere else. They are not mistakes in the deep ocean or monsters living outside the rules of biology. They are the result of a very specific set of conditions: volcanic rock, circulating seawater, dissolved chemicals, crushing pressure, cold background water, steep temperature gradients, and microbes that can build food without sunlight. That is what makes underwater volcanoes so strange. They create oases in a part of the planet where food is usually scarce, and they do it with energy that comes from the planet's interior rather than the surface. The animals gathered around them live in a world where the sun is irrelevant. Their food web begins in chemistry. Their bodies are shaped by hot fluids and toxic gases, by mineral crusts, by the brief life of event that may last for decades or collapse without warning. If the hidden ocean is what brings you here, subscribing keeps the next descent close. A hydrothermal vent begins with a crack. Seawater seeps down into the oceanic crust through fractures in basalt. Sometimes on a mid-ocean ridge where tectonic plates are spreading, sometimes along a volcanic arc, sometimes on a seamount where the seafloor has been built by eruption after eruption. As the water sinks, it is heated by hot rock and reacts with the minerals around it. It loses some chemicals, gains others, becomes more acidic, and strips metals from the crust. Then, because it is hot and buoyant, it rises again. When it comes back out into near freezing deep water, the dissolved minerals can no longer stay dissolved. They form particles. They darken the plume. They build chimneys one thin layer at a time. Some vent fluids can reach about 400 degrees Celsius or more, but at depth they do not boil in the familiar way, because the pressure is so high that water behaves differently. The result is one of the most extreme boundaries in the ocean: water hot enough to alter rock pouring into a sea only a few degrees above freezing. A vent field is not a single thing. It is a small landscape made from chemistry and time. There may be black smokers, where the plume is dark with metal sulfides. There may be white smokers, where lighter minerals make the flow pale. There may be diffuse vents, where warm water leaks through cracks and mats of bacteria spread across rock like pale felt. There may be dead chimneys too, hard mineral towers left behind after the plumbing below has shifted. To an ROV camera, all of this may fit into one crowded frame. To the animals, it is a map of survival. A crab that moves a few centimeters may pass from food-rich warm flow into cold water where the chemical supply fades. A tubeworm plume may extend into a mixing zone where oxygen and sulfide can both be captured. A shrimp may hold position in turbulence that would carry a weaker swimmer away. The entire habitat is built from gradients, and the gradients are sharp. Heat, sulfide, oxygen, acidity, and flow can change over the length of an animal's body. Here, the edge is not symbolic. It is where life has to stand. Before the late 20th century, scientists did not expect communities like these. The deep sea was already known to contain life, but most deep-sea food webs were understood as depending on material falling from above: dead plankton, fecal pellets, fragments of animals, the slow rain of organic matter often called marine snow. That still matters across most of the deep ocean. It feeds sediment communities, midwater animals, scavengers, and even distant seafloor habitats. But hydrothermal vents revealed another foundation. In 1977, cameras and the submersible Alvin helped document warm water venting and dense animal communities along the Galapagos Rift, about 2,500 meters below the surface. The discovery changed how scientists thought about where life could be abundant, because it showed that sunlight was not the only way to power a large ecosystem. The black water on the screen was not just scenery. It was evidence that the deep earth and the living ocean were connected more directly than anyone had understood. The discovery had the feeling of a contradiction. At the surface, life is usually tied to photosynthesis. Plants, algae, and cyanobacteria use sunlight to fix carbon, and most animals eventually depend on that work, even if they live far away from a leaf or a plankton cell. At vents, the first workers are microbes using chemical energy. Many oxidize hydrogen sulfide or methane or hydrogen, depending on the site, using those reactions to fix carbon dioxide into organic matter. That organic matter becomes the base of the food web. Some microbes live free on rocks and in mats. Some drift in the plume. Some live inside animals, not as passengers but as partners so necessary that the host cannot live without them. The alien quality of vent life begins here, at a scale too small for the camera to show clearly. The big animals around the chimney are visible because microbes are doing invisible work inside the dark water, inside tissue, inside mineral pores, and sometimes inside the crust itself. Giant tube worms make this relationship visible in a way that is difficult to forget. In the eastern Pacific, Riftia pachyptula can stand in dense white clusters around vents, each worm protected inside a long tube, each red plume reaching into the moving water. The red color comes from hemoglobin, and that blood-like molecule has to solve a problem that would be dangerous for most animals. It carries oxygen from seawater and hydrogen sulfide from vent fluid to symbiotic bacteria housed inside the worm's body, in an organ called the trophosome. The adult worm has no mouth, no stomach, and no intestine. It does not graze. It does not hunt. It farms chemistry inside itself, although even that phrase is too simple, because the bacteria are not a crop placed there by choice. The animal and the bacteria form a living system. The worm supplies raw materials. The bacteria fix carbon. The worm receives food from organisms it never sees. A tube worm colony can look still from a distance, like a bed of white reeds in the beam of the ROV. Up close, it is full of small decisions and physical responses. Plumes extend and withdraw. Crabs climb between tubes. Limpets graze on surfaces. Small fish turn at the edge of the warm flow. Scientists can describe the anatomy, chemistry, and symbiosis. But direct observation of daily life remains limited. The camera arrives for hours or days, not for the entire life of the vent. It sees the colony during one state of flow, one stage of succession, one weather pattern in deep water. It records what the lights reveal. It misses what happens behind the chimney, under the rocks, after the vehicle leaves. This is part of why vents keep their power as scientific places. They are not unknown because nobody has studied them. They are partly unknown because studying them is like trying to understand a city by visiting a few streets at night through the window of a machine. The phrase underwater volcano can make the habitats sound simple, as if every vent were just a chimney on a cone-shaped mountain. The real seafloor is more varied. Many vents occur along mid-ocean ridges where plates separate and new crust forms from rising magma. Others occur behind subduction zones or on volcanic arcs, where one plate dives beneath another and melts or releases fluids that feed volcanic systems. Some are associated with seamounts, which may be active, dormant, or extinct. The vent field may sit on fresh lava, collapsed crust, steep slopes, pillow basalts, or broken sulfide deposits. The geology controls the chemistry, and chemistry helps control the animals. A vent rich in sulfide may support different symbioses than a vent where methane or hydrogen is more important. A fast-spreading ridge may create short-lived but productive vent habitats. A slower or unusual system may persist differently. To say life lives around underwater volcanoes is accurate, but the volcano is not just a backdrop. It is the engine, the architecture, and sometimes the disturbance that erases the community it fed. The surface ocean above these places may be calm. A ship can sit over a vent field with ordinary waves moving under its hull, seabirds crossing the sky, crew members walking between winches and containers, while 2 or 3 kilometers below the hull the seafloor is warm enough to stain water black with minerals. The cable lowers a vehicle through the sunlit layer, then through blue water, then through the twilight zone where animals migrate at night and hide by day. Below that, the last ordinary color fades. Red disappears first. Then the water becomes a dark volume marked by instrument lights, depth readings, and drifting particles. The ROV does not enter as a natural animal. It carries lamps, cameras, thrusters, manipulator arms, sampling boxes, titanium housings, glass spheres, and a tether. Every image of this world is made through a technological interruption. The animals are seen because a machine has brought a brief artificial day to a place that normally has none. When the vehicle reaches the bottom, the vent may announce itself before the chimney appears. Sensors can detect a change in temperature. The water may shimmer where hot fluid mixes with cold seawater. Particles may thicken in the beam. A plume may rise above the seafloor like a dark, wavering column. Then the rocks come into view, and the camera begins to find animals. There may be mussels packed tightly against one another, shells open just enough to filter or to exchange with their symbionts. There may be clams, half buried near diffuse flow. There may be snails with mineralized scales in some regions, or limpets pressed to bacterial films. Vent animals are not distributed randomly around the chimney. They occupy chemical zones. The closest spaces may be too hot or too toxic. The farther spaces may be too cold or too poor in reduced chemicals. The living ring around a vent is often a compromise between nourishment and damage. Chemosynthesis does not remove danger from the habitat. It creates food inside a dangerous habitat. Hydrogen sulfide can be poisonous. Metal-rich fluids can be chemically harsh. Temperatures can shift quickly. Oxygen is necessary for many symbiotic reactions, but oxygen and sulfide usually come from different parts of the mixing water, so animals must position themselves where both can be reached. This is one reason vent life can be dense without being easy. The animals are not simply thriving because the vent is generous. They are holding themselves inside a narrow usable range. The same fluid that feeds their microbes can kill their tissues if the exposure is wrong. The same chimney that gives them attachment can break apart, clog, or stop venting. The vent community is an oasis, but it is an unstable oasis, made by the movement of hot water through cracked rock. When the plumbing changes, the living city can lose its foundation without moving at all. Mussels at vents and seeps show another way animals can use microbial partners. Some species host bacteria in their gill tissues, where the symbionts use chemical energy and provide nutrition to the host. In certain vent systems, mussel beds form thick layers across rock, creating habitat for smaller animals that live among the shells. The mussels themselves may filter particles too, depending on the species and conditions, so their lives can combine ordinary animal feeding with chemical symbiosis. Clams can do something similar, with large gills that house bacteria and bodies positioned to reach sulfide-rich sediments or fluids. To a camera, a mussel bed may look familiar at first, almost like a shallow water reef carried into darkness. Then the context changes it. There is no kelp, no sunlight, no green plant surface, no ordinary shoreline productivity. The shells are clustered around leaks in volcanic rock, and the food web running through them begins below their own feet. Shrimp can dominate some vent fields so completely that the first impression is movement. On the mid-Atlantic ridge, dense swarms of Rimicaris exoculata gather on active chimneys, their pale bodies stacked and shifting in the turbulent water. These shrimp are often described as blind, but they are not simply animals missing a sense. They have specialized light-sensing organs that may help them detect the faint glow or thermal cues associated with venting. Their bodies carry microbial communities, including bacteria associated with sulfur oxidation, and they feed in ways closely tied to these microbes and to the vent environment. A swarm can look chaotic in the RUV lights, thousands of small bodies pressed against the chimney, antennae crossing, legs scraping, shells flashing white. But it is an organized response to a rare energy source. Each shrimp is trying to remain near enough to the chemical flow to feed and far enough away to stay alive. The Pompeii worm lives closer to the heat than almost any animal people commonly imagine. Alvanella pompeiana builds tubes on the sides of active vent chimneys, placing parts of its body in very warm conditions while its head reaches into cooler water. The exact temperature tolerance of the animal has been debated and refined through study, because measuring the real conditions around a living worm is difficult. The flow is variable. The temperature changes across small distances. The animal's tube and microbial covering complicate the boundary between body and environment. Still, the basic fact remains remarkable. This worm lives on a chimney where heat, acid, and chemistry are intense enough that most animals could not function. It is not invincible. It is not a creature of lava. It is a polycate, an annelid worm, living at a sharp interface and using structure, behavior, and microbial association to survive in a place that seems almost designed to make animal life impossible. Vent life is often described through its most dramatic animals, but the community is broader than tube worms, mussels, shrimp, and Pompeii worms. There are scale worms moving across tubes. There are amphipods and copepods. There are snails grazing on microbial films. There are crabs and squat lobsters that feed, scavenge, and sometimes prey. There are fish hovering near the edge of the field, entering when the energy reward is worth the risk. There are octopuses in some vent-influenced or volcanic deep-sea settings, not always feeding directly on vent production, but using the hard seafloor and warm water in ways researchers are still studying. The vent is not a single food chain. It is a web of microbial production, grazing, symbiosis, predation, scavenging, larval settlement, competition for space, and chemical tolerance. Its animals may look alien because their habitat is extreme, but their lives still include the ordinary pressures of animal existence: finding food, avoiding being eaten, reproducing, growing, repairing damage, and dying. A dead chimney can be crowded with evidence of what used to happen. The mineral surface remains, but the living cover changes. Bacterial mats fade. Tubeworm tubes stand empty. Mussel shells gape. Crabs pick through what is left. When venting stops, the chemistry that supported the community declines, and many animals that depended closely on that flow cannot continue there. Some mobile animals leave if they can. Sessile animals remain attached to a place that no longer feeds them. The habitat becomes a record of interruption. In the deep sea, where many processes are slow, vents can be surprisingly temporary. A field may persist long enough for generations of animals to establish, or it may be reorganized by eruption, clogging, collapse, or shifting flow. This makes vent ecology different from the image of the deep sea as a stable, unchanging world. Around underwater volcanoes, disturbance is not rare. It is part of the system that creates the habitat in the first place. After an eruption, a vent field can be stripped back to rock and heat. Lava may bury old communities. Chimneys may topple. Animals that could not move quickly enough may be killed. Then, the rebuilding begins, not as a sentimental recovery, but as colonization. Microbes occupy fresh surfaces. Larvae arrive in currents or from nearby refuges. Limpets, worms, and other pioneers settle where chemistry and surface allow them. Over time, a new community develops, sometimes similar to the old one, sometimes different because the physical and chemical landscape has changed. Research after eruptions on ridges, such as the East Pacific Rise, has shown that larvae can colonize from surprising distances, though the exact routes, survival strategies, and current pathways remain active questions. The deep sea does not provide easy corridors. It provides moving water, patchy habitat, brief larval stages, chemical cues, and long gaps between suitable places. A vent field is an island, but the ocean around it is not empty. The island comparison is useful only to a point. Islands at the surface are surrounded by air or shallow water, and many animals can see their boundaries. Vent fields are islands of chemistry scattered along ridges and volcanic structures, surrounded by deep seafloor that may be cold, dark, and poor in the same reduced chemicals. A larval tube worm or mussel does not travel with a map. It drifts through water shaped by currents, turbulence, density layers, and plume chemistry. Some larvae may delay development. Some may respond to chemical signals near a suitable habitat. Some may fail to find a vent at all. Many likely die before settlement. The existence of a colony means that enough of them succeeded. It means that a tiny body, nearly invisible in the dark water, crossed a distance that would be enormous at its scale and arrived at a crack in the seafloor where chemistry, surface, flow, and time were all briefly right. Vents also show that the deep sea is not one habitat. The open midwater above them has its own animals, lanternfish, bristlemouths, siphonophores, squid, gelatinous predators, and planktonic forms moving through darkness. The abyssal plain around them may contain holothurians, brittle stars, worms in sediment, small crustaceans, and microbial communities fed largely by marine snow. A whale fall kilometers away would create another kind of island, based on fat, bone, and decay. A cold seep would support chemosynthetic life with different plumbing, often built around methane and sulfide leaking from sediments rather than volcanic heat. Hydrothermal vents belong to this larger deep-sea mosaic. They are not the whole deep ocean. They are one of its most concentrated expressions of energy, one of the places where the hidden rules of the seafloor become visible because the animals gather tightly enough for the camera to see them all at once. The background water around many vents is cold enough to slow ordinary metabolism, yet the vent itself injects heat into that cold environment. This creates not just a thermal boundary, but a biological one. Cold, deep water carries oxygen. Hot vent fluid carries reduced chemicals, but little usable oxygen. Life is most abundant where the two mix. The chemistry is not useful in isolation. Sulfide without oxygen is not enough for many symbioses. Oxygen without sulfide does not feed the bacteria that sustain the host. The vent animals live where separate worlds overlap. That overlap can be narrow enough to measure in centimeters and large enough, across a field, to sustain thousands or millions of animals. The camera shows a chimney and a crowd of white bodies. The scientific instruments show why they are crowded there. Their abundance is written in gradients: oxygen here, sulfide there, heat rising, cold water entraining, microbes fixing carbon at the line where incompatible conditions briefly meet. Pressure is always present, even when the narration focuses on heat. At 2,500 meters, the pressure is roughly 250 times the pressure at the surface. At deeper vent fields, it is higher still. This pressure changes the behavior of water and gases, changes how equipment must be built, and affects the bodies of animals adapted to live there. A human diver cannot simply enter this place. A shallow water animal cannot simply descend into it. ROV housings are sealed and engineered. Glass spheres and titanium parts resist compression. Samples brought to the surface may change as pressure falls. Some animals lose shape after recovery. Some microbes behave differently outside their native conditions. Some chemical compounds oxidize or precipitate before they can be fully understood. For this reason, the best evidence often comes from in situ instruments, from measurements made in place, because the vent is not only a location. It is a physical state that changes when removed from depth. The name black smoker is both descriptive and misleading. It looks like smoke, but it is not smoke. It is a suspension of fine mineral particles forming as hot, metal-rich water meets cold seawater. Iron sulfides and other minerals darken the plume, and over time, those minerals can build the chimney walls themselves. The chimney is both a structure and a record of flow. Its shape reflects where fluid has moved, where minerals have precipitated, where collapse has happened, and where animals have attached. A manipulator arm can break a piece from the side and place it in a sampling box, and that fragment may hold chemical history in layers. Under a microscope or in a lab, the rough black surface becomes more than a dramatic image. It becomes evidence of reactions between water and rock, of metals moving through the crust, of microbial colonization, and of the way the planet builds habitats out of its own interior heat. White smoker systems can feel quieter on camera, though they are no less strange. Instead of a dense black plume, the fluid may emerge pale or shimmering, rich in different minerals and often at lower temperatures than the hottest black smokers. Some alkaline hydrothermal systems, such as the Lost City field on the Mid-Atlantic Ridge, are driven not by the same style of magmatic heat as black smokers, but by reactions between seawater and mantle rock in a process called serpentinization. These reactions can generate hydrogen and methane, creating chemical conditions that interest microbiologists, geochemists, and astrobiologists. The towers at such sites can be light-colored carbonate structures, rising from the seafloor like mineral architecture built by water-rock reactions. The animals there may not form the same dense displays as some sulfide-rich vents, but the chemistry is important because it shows that underwater volcanic and hydrothermal systems are not all variations of one scene. They are many ways for rock, water, and life to meet. Alien life around underwater volcanoes is also a phrase that points outward, toward other worlds. Icy moons such as Europa and Enceladus are interesting to scientists partly because they may have liquid oceans beneath ice, and those oceans may interact with rock below. If water, rock, heat, and chemical disequilibrium exist together, then some of the ingredients that make Earth's vent ecosystems possible might exist elsewhere too. That does not mean tube worms are waiting under alien ice. It does not mean shrimp or crabs belong in the prediction. The lesson is more restrained and more important. Life on Earth has shown that sunlight is not required for every productive ecosystem. Microbes can use chemical energy. Animals can build bodies around microbial partners. Complex communities can form in darkness if energy and chemistry are available. The vents do not prove life beyond Earth. They expand the range of places where the question is scientifically reasonable. The search for life elsewhere often returns to the smallest organisms at the vent, because microbes carry the deepest implications. A tube worm or shrimp is visually compelling, but the microbial metabolism is the foundation. Chemolithoautotrophic bacteria and archaea can use reduced compounds from vent fluids as electron donors and fix carbon into biomass. Some live attached to surfaces. Some live as symbionts. Some occupy spaces inside porous rock. Some are carried in hydrothermal plumes away from the vent. Their diversity varies with temperature, pH, mineralogy, and fluid chemistry. A sample of vent water may look like a vial of dark liquid. A scrap of mineral crust may look inert. Yet both can contain communities adapted to chemical boundaries that would be invisible to the unaided eye. When scientists sequence DNA from these samples, they are not just naming species. They are reading different ways of making a living from the Earth's interior. One of the most recent changes in how scientists picture vent habitats came from work beneath the visible seafloor. By lifting pieces of lava crust near hydrothermal vents on the East Pacific Rise, researchers found animals such as tube worms, snails, and bristle worms living in shallow cavities below the surface of the seafloor. This did not mean the entire ocean crust was full of large animals. It meant the vent habitat extended into spaces that previous cameras could not see, connecting the surface colony with the subsurface circulation of warm fluids. The image is simple and unsettling. an rough arm prying up a lava plate, a dark cavity underneath, and living animals where the seafloor had seemed solid. It suggests that part of the vent community may not only live around the chimney, but under the volcanic pavement that carries the hot water upward. That hidden layer changes the meaning of the visible colony. A tubeworm forest above the seafloor is not necessarily the whole organismal city. It may be the exposed part of a larger habitat linked by cracks, cavities, and moving water. Researchers have proposed that larvae may move through or settle within these subsurface spaces, but the details remain incomplete. How far animals can travel under the crust, how often this happens, how widespread it is across vent systems, and how it affects recovery after disturbance are questions that require more dives, more sampling, and more patience. This is a familiar pattern in deep-sea science. The first discovery reveals a scene. Later work reveals that the scene had an underside. Then the underside becomes its own field of questions. The seafloor that looked like a boundary becomes a roof. The animals that seemed to occupy the surface become evidence of a habitat extending into the rock below them. Life around vents is dense, but it is not protected from the violence that creates it. Submarine eruptions can pave over communities with lava. Earthquakes can open new cracks and seal old ones. Chimneys can grow, clog, fracture, and collapse. Hydrothermal flow can shift from one part of a field to another, turning a living zone into a mineral ruin while another begins to warm nearby. In some places, scientists have returned to the same vents over years and watched the community change. New lava becomes colonized, young tubeworms establish, mussel beds expand, predators arrive, old tubes decay. Succession at vents is not identical everywhere because the species pool, chemistry, and geology differ from region to region, but the principle is clear. These ecosystems are dynamic on timescales short enough for human instruments to record. In the deep sea, where many things seem slow, underwater volcanoes can make ecology move quickly. The animals have to reproduce into that uncertainty. A sessile adult tubeworm cannot leave when the vent begins to fail. A mussel cannot swim across the ridge to another active chimney. Their offspring must do the traveling. Many vent animals release larvae into the water, and those larvae enter a deep ocean where suitable habitat is rare and patchy. The larvae may feed or rely on stored energy, depending on species. They may remain in the water column for days, weeks, or longer. They may encounter plumes that provide chemical cues. They may settle on rock where microbial films are already forming. Most of them will not become adults. That is not unusual in the sea. What is unusual is the kind of address they must find, not a reef lit by sun, not a sandy bottom, not a kelp forest, but a leaking volcanic seam where the chemistry is strong enough to feed symbionts and gentle enough not to kill the host. The camera sometimes catches young stages only as hints. Small tubes on fresh basalt, tiny snails on a mineral surface, a thin microbial film where animals will later gather. These are not dramatic images in the ordinary sense, but they may be among the most important. They show a vent field before it becomes visually crowded. They show the process by which chemical energy is converted into habitat. They show that a living community does not appear all at once. It assembles, with microbes first, then grazers, then larger animals, then predators and scavengers following the concentration of bodies. The exact order varies. The timing varies. A vent field is not following a script. It is responding to flow, chemistry, larvae, competition, and chance. The result can look inevitable once a colony is mature, but in its early stages it is full of failed settlements and narrow openings that only some organisms manage to use. A vent crab does not care that humans call the scene alien. It moves because there is food, because there are rivals, because the water around it carries signals its body can use. Crabs and squat lobsters may scavenge dead animals, graze bacterial growth, or prey on smaller vent organisms. Their claws and legs make them seem more familiar than tube worms or symbiotic shrimp, but they too are shaped by the conditions of the field. They must tolerate chemical exposure, unstable footing, and thermal variation. They must find enough energy in a patchy system. Around a dense colony, death becomes part of the food web quickly. A dead worm or mussel does not lie untouched for long. Scavengers arrive. Microbes continue the breakdown. Shells and tubes become surfaces for other organisms. The vent creates life by chemistry, but once bodies exist, they enter the same cycles of injury, predation, decay, and recycling found everywhere animals live. Fish around vents are often less famous than the invertebrates, but they matter because they connect the vent to the wider deep-sea food web. Some species are strongly associated with vent habitats, while others visit from surrounding areas. Fish can hover at the edge of the warm flow, feed on crustaceans or worms, and move between patches more easily than sessile animals. Their presence reminds us that vents are not sealed worlds. Energy produced at the chimney can move outward through predation and scavenging. Animals that feed near vents may themselves be eaten away from vents. Larvae move through the water column. Plumes carry particles and microbes above the field. A vent is concentrated, but it is not isolated in every sense. Like a whale fall, a seep, or a seamount, it is a place where deep-sea energy becomes locally intense and then leaks into the surrounding ocean through bodies, currents, and time. The ROV's lights can make vent animals look pale and ghostly, but color at depth is not what it seems in footage. Red light from the surface is gone long before these depths, and many animals live without the kind of full-spectrum illumination humans bring with them. The red plume of a tube worm appears red because the vehicle lights restore wavelengths that the deep ocean itself does not provide. White shrimp may look bright because they reflect artificial light. Mineral surfaces may flash gold, black, gray, and orange under lamps, even though in natural darkness those colors are not displayed to a human eye. This is one of the quiet distortions of deep-sea exploration. The scene becomes visible by being altered. Still, the alteration is useful. Without the lights, the camera would show almost nothing. With them, we can see bodies, tubes, shells, motion, and the boundaries of a world that normally exists without ordinary sight. Not every strange deep-sea animal near volcanic terrain is dependent on vents. This matters because the title can tempt the imagination to gather every unusual organism into one category. Some animals live on seamounts because currents concentrate food there. Some live on volcanic slopes because hard rock provides attachment in a world of sediment. Some deep-sea corals, sponges, brittle stars, and fishes may occupy old lava or inactive mineral surfaces without using chemosynthesis directly. Others live near active flow and depend on microbial production. The difference is ecological, not just visual. A sponge garden on a seamount, a coral thicket on a ridge, and a tubeworm colony at a black smoker all belong to the deep ocean, but they do not work the same way. To understand alien life around underwater volcanoes, the important question is not whether the animal looks strange, it is what energy source supports it, what chemistry it tolerates, and how closely its life is tied to the volcanic system. Seamounts can rise thousands of meters from the seafloor without breaking the surface. Some were once volcanoes active enough to build mountains underwater, and many become places where currents accelerate, lift nutrients, and carry food past hard surfaces. A camera moving along a seamount slope may find corals branching into the current, sponges shaped like cups or fans, crinoids holding feathery arms into the flow, and fish resting where the current breaks. This is another kind of volcanic deep sea life, less chemically extreme than a hydrothermal vent, but still shaped by rock made from Earth's interior. The animals here may depend on particles from the water column rather than vent chemicals. Their food may come from plankton production far above, sinking debris, or small animals carried by currents. Their alien quality comes from depth, slowness, suspension feeding, and form, not necessarily from chemosynthesis. The volcano made the mountain. The mountain reshaped the current. The current made feeding possible. At an active vent, time is compressed by heat. Minerals precipitate quickly. Chimneys grow. Bacterial mats can expand across new surfaces. Animals that can settle and grow fast have an advantage because the habitat itself may not last. Giant tube worms are famous partly because they can grow rapidly compared with many deep-sea animals, using abundant chemical energy through symbiosis. This contrasts with the slow growth often associated with cold, food-poor abyssal environments. The deep sea is often described as slow, but that description is not universal. Vents can be productive, crowded, and changing. They sit inside the deep ocean, but they do not follow every deep ocean pattern. The lesson is not that the deep sea is slow or fast. It is that the deep sea contains many clocks. A coral on an old seamount may live for centuries. A vent chimney may change between expeditions. A larva may have only a brief window to find the chemical address that can become its life. The animals that depend on vents must also solve the problem of toxicity. Hydrogen sulfide interferes with aerobic respiration in many animals, binding in ways that can prevent cells from using oxygen. Vent organisms have biochemical adaptations and symbiotic arrangements that allow them to handle compounds that would be dangerous elsewhere. Tubeworm hemoglobin can bind both oxygen and sulfide and deliver them without poisoning the worm's own tissues in the usual way. Some mussels and clams manage sulfide through specialized tissues and bacterial partners. Shrimp and worms live with microbial communities on or inside their bodies that transform chemical conditions. These adaptations are specific. They are not evidence that vent animals are immune to all extremes. A change in concentration, temperature, or flow can still be lethal. Their success comes from fitting a narrow chemical world with unusual precision, not from being beyond vulnerability. If a manipulator arm takes a tubeworm, the animal that arrives on deck is no longer exactly the animal from the vent. Pressure has changed, temperature has changed, chemistry has changed. The plume is gone. The symbionts are removed from the gradients that made their work possible. Scientists can still learn from the specimen. They can examine tissue, bacteria, blood chemistry, growth rings, tube structure, and isotopes. They can compare individuals from different vent fields. They can preserve pieces in ethanol, freeze samples, or keep some organisms under pressure in specialized systems. But the act of collection always has limits. A vent animal is not only a body, it is a body in flow, in pressure, in chemical exchange, in a community. The more completely researchers can study it in place, the closer they come to the actual life of the animal. A control room during a dive is a strange human shoreline. There may be no view of the sea outside, only monitors, sonar displays, navigation charts, depth readouts, and voices calling out observations. Someone marks a temperature anomaly. Someone notes shrimp density. Someone asks the pilot to move the arm slowly toward a sample. The vehicle's lights brighten mineral walls that no human eye could reach unaided. In the screen glow, scientists argue gently over identification, whether a cluster is mussel or clam, whether the shimmering water marks diffuse flow, whether a patch of pale material is bacterial mat or mineral coating. This is not a heroic conquest of the deep. It is partial attention under difficult conditions. The ROV can hover, but currents push it. The arm can sample, but it can also break fragile structures. The cameras can record, but only where they are pointed. Knowledge arrives frame by frame, with uncertainty visible in the way people speak while the vehicle moves. The first vent discovery in 1977 is often treated as a clean moment, but science rarely stays clean after discovery. Once vents were known, researchers had to ask how common they were, how different regions compared, how animals dispersed between fields, how microbial metabolisms varied, how vent chemistry changed over time, and how these ecosystems fit into global ocean processes. The answers did not come from one dive. They came from repeated expeditions, long-term observatories, chemical sensors, genetic studies, larval traps, manipulative experiments, seafloor mapping, and comparisons across oceans. A black smoker filmed once can astonish. A black smoker revisited year after year can teach. It can show growth, collapse, replacement, colonization, and decline. It can turn a mysterious scene into a changing habitat with measurable processes, while still remaining difficult enough that each return reveals something missed before. The Galapagos Rift, the East Pacific Rise, the Mid-Atlantic Ridge, the Juan de Fuca Ridge, the Indian Ocean ridges, and vent systems in the Western Pacific all show that hydrothermal life is not one uniform community repeated globally. Different oceans have different species pools. Barriers between ridge systems can limit dispersal. Chemistry varies. Depth varies. Geological setting varies. Some fields are dominated by tube worms, some by mussels, some by shrimp, some by snails or other assemblages. The same broad strategy, using chemical energy through microbes, can produce different visible worlds depending on location. This is similar to how forests on land can all rely on photosynthesis while still differing in their trees, insects, birds, and soils. The vent principle is global, but the communities are local. Each field is a version of life around underwater volcanoes written in its own chemistry and evolutionary history. The giant tube worm became an icon because it gave the vent discovery a body large enough to symbolize the whole system, but a symbol can distort if it becomes too dominant. Riftia is not present at every vent. Many vent animals are small, hidden, regionally restricted, or hard to identify without sampling. Microbes, which make the system possible, rarely get the same attention because they do not tower in the lights. A good vent story has to keep returning to scale. A white worm tube may be a meter long. A bacterial cell may be a few micrometers across. A vent field may cover only a small area of seafloor. The ridge system that hosts such fields may stretch across ocean basins. The processes that supply heat may reach into the mantle over geological time. Life is concentrated in small spaces because planetary scale forces have opened a chemical door. The chimney itself is not alive, but it behaves almost like a growing structure. Minerals deposit on its walls as hot fluids meet cold seawater. Channels open and close inside it. Pressure forces fluid through weak points. Pieces break away. New spires rise from old bases. Animals settle on the surface and alter the flow around them. Microbial films coat mineral faces. Over time, the chimney becomes both geology and habitat, a tower built by reaction and occupied by organisms that depend on those reactions continuing. It can also fail suddenly. A wall can collapse under its own weight. A vent orifice can clog. A burst of hotter flow can change the usable zone. A colony attached to the chimney is living on infrastructure that the planet is still actively making and unmaking. This is not a reef in the shallow tropical sense, built slowly by animals in sunlight. It is a mineral scaffold formed by hot water. The animals around vents also complicate the old idea that deep sea life is always sparse because food is scarce. Most of the deep ocean is indeed limited by the amount of organic matter sinking from above. A camera can travel over sediment for long stretches and see only scattered organisms: a sea cucumber, a brittle star, tracks in mud, a fish resting close to the bottom. Then the same vehicle can reach a vent and find dense life packed into a small area. The difference is not depth alone, it is energy. Where food arrives only as a diluted rain, animals are spread thinly. Where chemical energy emerges from the crust and microbes can use it, biomass can concentrate. The deep ocean is not empty, but it is uneven. Its abundance gathers where conditions allow it. Around underwater volcanoes, one of those conditions is a crack that lets the inside of Earth feed the surface of the seafloor. A vent field has sound in a physical sense, but not in the way the script can easily use. Water moves. fluids rush. Animals scrape and crawl. The ROV thrusters hum through the structure. Yet the footage often feels quiet because the camera removes most of the sensory world. Viewers see drifting particles, a plume, a crab, a worm forest. They do not feel the pressure, smell the sulfide, sense the turbulence through a shell, or read chemical gradients through bacterial metabolism. This is why narration has to be careful with atmosphere. Darkness is not a mood to the animals. It is a lighting condition. Heat is not drama. It is a variable. Sulfide is not menace. It is both danger and resource. The vent is strange enough when treated plainly. It does not need to be turned into fantasy. The real habitat, with its narrow tolerances and dense dependencies, is more interesting than a supernatural version of it. Deep-sea hydrothermal vents are often found at depths between about 2 and 4 kilometers, though specific fields can be shallower or deeper. At these depths, the pressure changes the boiling point of water and allows superheated fluids to remain liquid as they exit the seafloor. The surrounding seawater may be near 2 degrees Celsius. The vent fluid may be hundreds of degrees. Between them is a turbulent mixing zone where microbial metabolism and animal symbiosis become possible. A thermometer moving through the flow would not record one stable number. It would jump and flicker as hot and cold filaments passed the probe. An animal living there experiences that variability constantly. Its body is not sitting in a simple bath of warm water. It is inside a moving boundary where chemistry and temperature are braided together by turbulence. The mineral wealth of vents has always drawn another kind of attention. Hydrothermal systems can form deposits rich in copper, zinc, gold, silver, and other metals, because the same fluids that feed microbes also transport elements leached from rock. Ancient deposits formed by hydrothermal processes are mined on land. Active seafloor massive sulfide deposits have attracted interest as possible targets for deep-sea mining. This raises difficult questions, because active vents are not just mineral deposits. They are living habitats, sometimes with species found nowhere else. Disturbing a vent field could remove the hard structure, alter flow, create sediment plumes, destroy animals, and change the conditions needed for recovery. The scale of impact would depend on the site and activity, but the concern is straightforward. Around underwater volcanoes, geology and biology are too tightly joined to treat the rock as empty. Not all vents are equally vulnerable in the same way, and not all inactive sulfide deposits support the same communities as active vents. Some inactive deposits may still host animals on hard substrate, but without active chemosynthetic flow, their ecology differs. Active vents, where chemical energy supports endemic communities, raise special concerns because disturbance can remove both organisms and the physical chemical conditions they require. Scientists studying conservation often have to map vent fields carefully, distinguish active from inactive structures, identify species, understand connectivity between populations, and estimate how quickly or whether recovery could occur after disturbance. The uncertainty cuts both ways. It does not prove that every impact would be permanent everywhere, but it also does not allow confidence that recovery would be quick or complete. In a habitat built from rare chemistry and patchy distribution, losing one field may mean losing more than a scenic cluster of chimneys. It may mean cutting a node in a deep, poorly understood network. The phrase life without sunlight can sound like a complete separation from the surface world, but vents are not entirely cut off from the rest of the ocean. Oxygen in deep water ultimately comes from photosynthetic life and atmospheric exchange far away. Some animals near vents may also eat particles that originated above. Predators can move between vent and non-vent habitats. Larvae drift through the same deep water that carries marine snow. Chemosynthesis creates a local source of carbon, but the vent community still exists inside the larger ocean system. Its water masses have histories, its oxygen has a source, its currents connect regions, its animals interact with the surrounding deep sea. This makes the vent more, not less, Interesting. It is not a sealed alien planet at the bottom of ours. It is a place where Earth's surface and interior meet through the ocean, where sunlight-derived oxygen and rock-derived chemical energy are brought together in living tissue. A tube worm plume is one of the clearest images of that meeting. The plume extends into oxygenated seawater. The worm's lower body sits protected in the tube near sulfide-rich flow. Hemoglobin carries both substances inward. Bacteria in the trophosome use chemical energy to fix carbon. The animal grows without a mouth. If you hold that sequence in mind while looking at the ROV footage, the red plume stops being only beautiful. It becomes an instrument. It is the part of the worm exposed to the mixing zone, the living interface between two fluids. A fish swimming past may see only a forest of red tips. A crab may see climbable tubes and potential food. A scientist sees anatomy solving chemistry. The alien feeling comes from how unfamiliar the solution is, not from the animal being outside biology. It is biology using a route that surface animals rarely need. Vent animals can also be fragile in ways that footage hides. A white tube may look hard and simple, but inside is soft tissue dependent on continuous flow. A mussel shell can resist pressure and predators, but the animal inside relies on bacteria and exchange across delicate gills. A shrimp swarm can look abundant enough to be inexhaustible, but abundance at one chimney may not mean security across a whole ridge system. Many vent species have limited ranges, specialized habitat needs, or dispersal challenges. Some are known from relatively few fields, others may be widespread along connected ridges. The pattern varies. That variation is the point. To protect or understand vent life, scientists cannot treat it as one global mass of strange animals. They have to study populations, currents, genetics, chemistry, and disturbance history. The more closely the habitat is examined, the less it looks like a single alien world, and the more it looks like many precise worlds, each with its own constraints. The ROV arm often moves slowly around vents because almost everything is both valuable and breakable. A careless touch can snap a tube, crush a chimney flange, stir sediment into the cameras, or scatter animals before behavior is recorded. Sampling is necessary, but it is never neutral. Scientists may choose a small piece of chimney, a few animals, a water sample, a microbial mat, a temperature profile. Each sample removes something and reveals something. Ethical fieldwork in these habitats often means taking enough to answer real questions and no more than needed, documenting context with images and measurements, and recognizing that the deep sea is not a limitless storage room for human curiosity. The machine's arm is a strange extension of human touch, reaching into a place humans cannot enter directly. Every movement carries the tension between wanting to know and wanting the habitat to remain intact after the lights move on. The chemistry of vents can be described in equations, but the animals make it physical. Hydrogen sulfide becomes a red plume and a trophosome. Methane becomes muscle tissue at some seeps and vent-related systems. Hydrogen becomes microbial energy in alkaline hydrothermal settings. Iron, copper, zinc, and sulfur become chimney walls. Calcium carbonate becomes pale towers at places like Lost City. These transformations are not metaphors. They are matter changing form through reactions, then entering bodies. A carbon atom fixed by a bacterium may become part of a worm, then part of a crab, then part of microbial decay, then dissolved again in seawater. The vent is not separate from the planet's cycles. It is one of the places where those cycles are unusually visible, because rock chemistry enters biology over a distance small enough for a camera to frame. The Lost City hydrothermal field deserves special caution, because it is often pulled into origin of life stories. Its alkaline fluids, hydrogen production, and carbonate towers make it scientifically important, and systems like it are relevant to hypotheses about how early metabolism might have emerged on Earth. But no modern vent field is a preserved copy of life's beginning. Today's microbes are the products of billions of years of evolution. Today's oceans have oxygen histories, biological feedbacks, and chemistry unlike the early Earth in many ways. The value of these systems is not that they let us watch life begin again. It is that they show how water-rock reactions can create chemical gradients, catalytic surfaces, and energy sources that may have mattered for prebiotic chemistry. They offer clues, not a time machine. The mineral towers are real structures in the present ocean, and their connection to life's origin remains a scientific question, not a finished story. If the camera pulls back from a black smoker, the crowded scene becomes very small. The chimney shrinks into a dark shape on a ridge. The tube worms and shrimp become pale texture. The plume becomes a stain moving upward. Around it spreads ordinary deep seafloor, dark, cold, and sparsely inhabited by comparison. Pull back farther, and the entire vent field becomes a point along a ridge thousands of kilometers long. Pull back again, and the ridge becomes one seam in the global system of plate tectonics. The scale shift matters, because vents are both tiny and planetary. The living community may occupy a small patch. The forces that make it involve plate movement, mantle heat, seawater circulation, and ocean chemistry. A crab walking over a tubeworm tube is participating in a habitat made by tectonics. But the crab does not need to know that. It only needs the next step to hold. The word volcano usually brings images of lava fountains, ash clouds, and glowing rivers of rock. Underwater, volcanism behaves differently because pressure, water, and rapid cooling change the form of eruption. Lava can emerge as pillow basalts, rounded lobes formed as molten rock meets cold seawater and skins over. It can form sheet flows across the seafloor. It can build seamounts, ridges, cones, and collapsed terrains. Explosive activity can occur in some settings, but much of the volcanic seafloor is built quietly from the perspective of anyone at the surface. Quiet does not mean harmless. Fresh lava can erase a vent community. New cracks can create another. The same volcanic system that supports chemosynthetic animals can destroy them without warning. Life around underwater volcanoes is therefore not a story of safety in an oasis. It is a story of organisms using a dangerous energy source, because it is also an abundant one. At many vents, the first visible color is not from animals, but from microbes. Mats can appear white, yellowish, orange, or gray, depending on the organisms and minerals involved. These mats may coat surfaces where diffuse flow provides reduced chemicals at tolerable temperatures. Grazers feed on them. Larger animals settle near them. Microbial films can influence larval settlement by changing the texture and chemistry of rock surfaces. A patch of white fuzz on basalt may look minor beside a forest of tubeworms, but it can represent the early stage of a community or an active zone of chemical conversion. It is the food web becoming visible before the animals dominate the frame. The deep sea often hides its foundations. At vents, the foundation sometimes appears as a pale skin over black rock, thin enough to scrape away, important enough to feed everything that follows. A vent snail moves through a world of sharp surfaces. Mineral grains, bacterial films, shell edges, broken tubes, and chimney crust form the ground beneath it. Some vent snails graze microbial growth. Some host symbionts. Some have unusual shells or body coverings linked to the mineral-rich environment. The famous scaly-foot gastropod from Indian Ocean vents incorporates iron sulfide minerals into parts of its sclerites, a feature that made it one of the most striking examples of mineralized animal armor. It is tempting to treat such an animal as fantasy, but its armor is not decoration. It is a biological structure shaped by chemistry, predation, and habitat. The snail is still a snail, moving slowly, feeding, reproducing, and trying not to be eaten. Around vents, even an ordinary action like crawling can involve materials and conditions that seem borrowed from geology more than zoology. Predation at vents is easy to overstate because the setting already feels severe. a crab eating a worm, a fish striking at shrimp, or a snail being pried from a surface can look like the whole meaning of the place if the footage catches it. But most of the time, animals are not performing dramatic scenes. They are holding position, grazing, filtering, extending plumes, scraping surfaces, cleaning appendages, or simply remaining within the usable flow. Predators matter because they move energy through the food web and shape behavior. They are not villains. A vent crab is also vulnerable when young. A fish must spend energy to hunt and avoid harmful water. A shrimp swarm is both abundant and exposed. The more accurately these animals are described, the stranger they become in the right way, not as monsters in darkness, but as living organisms solving ordinary problems under extraordinary conditions. The black water above a vent is not empty space. Hydrothermal plumes can rise hundreds of meters or more above the seafloor before spreading laterally, carrying heat, chemicals, minerals, and microbes into the deep ocean. Scientists can detect vent fields by searching for these plumes, following chemical anomalies, temperature signals, optical backscatter, or particles in the water column. Sometimes the first sign of a vent is not a chimney on camera, but a sensor trace in water far above it. A research ship may tow instruments through darkness, watching for a slight change that points toward an unseen source. The seafloor remains hidden, but the plume betrays it. In that sense, underwater volcanoes announce themselves through the water before they are seen. Their chemistry rises like a message, diluted by currents, still strong enough for instruments to follow back toward the crack where life is concentrated. Vent plumes also complicate the boundary of the ecosystem. The densest animals remain near the flow, but microbes and chemical products can travel away from the chimney. Some plume microbes may continue metabolic activity as the plume disperses. Particles may settle onto surrounding sediments. Animals outside the main vent field may encounter plume-derived material. This does not mean the entire surrounding seafloor becomes a vent community. The strongest biological dependence remains close to the source, where chemical energy is concentrated. But the influence of the vent is not a perfect circle drawn around the chimney. It is shaped by currents, buoyancy, mixing, particle size, microbial growth, and seafloor topography. A vent field is local, but its edges are dynamic. The camera likes boundaries because it sees where the animals thin out. Chemistry is less obedient. It moves. The deep ocean around vents contains its own nightlife. Above the seafloor, small crustaceans and fish may rise and fall in daily migrations tied to surface light, even though the vent itself is beyond sunlight. The Great Vertical Migration moves enormous biomass through the water column each night, as animals rise to feed in shallower waters and descend by day. Some of that life eventually becomes marine snow. Some becomes prey for larger animals. Some falls as carcasses. This rain from above continues even near vents, mixing the ordinary deep sea economy with the chemical economy of the volcano. A vent field is therefore not a pure laboratory of chemosynthesis. It is a habitat where several energy pathways can overlap. The alien life around the chimney still lives on the same planet as lanternfish, plankton, squid, and falling debris. The strangeness is real, but it is threaded into the rest of the ocean. Oxygen minimum zones can intersect with volcanic and hydrothermal regions in ways that further shape animal life. Where oxygen is low, animals must adjust metabolism, behavior, or habitat use. Some microbes thrive in chemical conditions that would stress larger animals. Vent communities already depend on the meeting of reduced chemicals with oxidants, so oxygen availability matters intensely. A small change in background water chemistry can alter the usable space around a vent. If oxygen becomes less available, animals that rely on aerobic processes or oxygen sulfide exchange may be compressed into narrower zones or forced into different behavior. The deep sea is not immune to broader ocean change. Warming, deoxygenation, acidification, and human disturbance can all interact with habitats that already operate near physiological limits. The animals around underwater volcanoes may seem adapted to extremes, but adaptation to one extreme does not mean freedom from all others. The idea of alien life often emphasizes resemblance to another planet, but perhaps the more important resemblance is to another Earth. Vents show a version of this planet that does not match the surface-centered story humans usually tell. Here, mountains are hidden underwater. Volcanoes breathe through cracks rather than through red craters. Food begins in minerals and microbes. Animals grow without mouths, carry bacteria as organs of nutrition, or swarm around hot fluids in darkness. The system is not magical. It follows thermodynamics, geochemistry, evolution, and ecology. Its strangeness comes from a different arrangement of familiar principles. Energy flows downhill through chemical reactions. Organisms capture some of it. Bodies grow. Predators follow. Communities assemble and break apart. To call it alien is to admit that Earth is larger than the version of Earth made by daylight, air, soil, leaves, and human senses. The animals themselves are often classified within familiar groups. Tube worms are annelids. Vent shrimp are crustaceans. Mussels and clams are bivalves. Crabs are decapod crustaceans. Snails are gastropods. Scale worms are polychaetes. Fish remain fish. This matters because the alien appearance of vent communities does not require alien ancestry. Evolution used existing lineages and pushed them into new relationships with microbes and chemistry. A mussel at a vent shares broad body plans with mussels in shallower water, but its gills may house symbiotic bacteria. A worm at a vent belongs to a wider animal lineage, but the adult may lack a digestive tract and depend completely on internal microbes. The animals are familiar and unfamiliar at the same time. That tension is part of their power. They show that ordinary animal groups can become extraordinary when the habitat asks different questions. A specimen jar in a museum can make vent life look smaller than it felt on the seafloor. A shrimp preserved in alcohol loses the swarm. A piece of tubeworm tube loses the black plume, the red plume, the flow, the crab climbing between neighboring tubes. A snail shell loses the surface it moved across. Yet preserved specimens are essential. They allow anatomy to be studied, species to be described, DNA to be compared, and future scientists to re-examine material with tools that did not exist when the animal was collected. Much of deep-sea knowledge depends on this partnership between fleeting footage and durable specimens. The video shows the animal in place. The specimen reveals internal structure. The sequence data traces relationships. The chemistry explains energy. None of these alone is complete. Together, they make a partial but growing portrait of life around underwater volcanoes. Some of the most important vent animals are recognized not by beauty, but by absence. The adult Riftia tubeworm has no mouth. That absence is a clue, a negative space in anatomy that forced scientists to ask how the animal lived. No mouth means no ordinary feeding. No gut means no ordinary digestion. The answer was not that the animal somehow needed no food, but that it had outsourced food production to symbiotic bacteria housed inside its own body. This is why anatomical details matter. A missing organ can reveal an entire ecosystem. A gill enlarged beyond ordinary expectation can point to bacterial symbiosis. A reduced eye can suggest life organized around senses other than vision. A mineralized body covering can reveal a relationship with local chemistry. In the deep sea, evidence often begins as a strange feature in a specimen, then widens into a question about habitat, behavior, and energy. The relationship between vent animals and bacteria is not always inherited in the same way. Some symbionts may be acquired from the environment during development. Others may be transmitted more directly depending on host group, although mechanisms vary widely. For tubeworms like Riftia, evidence indicates that larvae begin without their symbionts and acquire them from the environment, which means the bacteria must also exist outside the adult worm at some stage. This makes the surrounding habitat even more important. The vent is not only feeding adult animals, it is maintaining microbial populations that future hosts may need. A young worm settling on rock must not only find the right chemistry and surface, it must also enter a microbial world where the partners required for adult life are available. The animal's future depends on organisms too small to see, living in water, on rock, or in the hidden spaces around the vent. To live at a vent is to live with instability in the body as well as the landscape. Symbiosis is powerful, but it requires exchange. The host must deliver chemicals, remove waste, maintain tissues, and regulate conditions for microbes that are both useful and potentially demanding. The microbes must fix carbon efficiently enough to support the host. If flow declines or chemistry shifts, the partnership can weaken. This is not unlike other symbioses in principle, such as corals and photosynthetic algae in shallow water. But the energy source and environment are very different. Coral symbiosis is tied to light. Vent symbiosis is tied to reduced chemicals and oxidants. Both can build abundant communities. Both can fail when environmental conditions move outside tolerable limits. The comparison helps only if it is kept precise. The vent is not a coral reef in darkness. It is its own kind of animal microbe construction, made around volcanic chemistry. The boundaries of vent life can be seen in the animal spacing. Tube worms may cluster densely where diffuse flow is strong, then thin out abruptly where sulfide becomes unavailable or conditions become too harsh. Mussels may form beds around cracks, but leave bare mineral surfaces nearby. Shrimp may crowd active chimneys and avoid dead structures. Ecologists can map these distributions and compare them with temperature, chemistry, and flow. The map becomes a biological reading of invisible gradients. Where the animals gather, something in the water is right. Where they are absent, something is missing or excessive. The ROV camera sees bodies. The sensors reveal why the bodies are there. In a habitat without sunlight, location is not about shade or exposure in the ordinary sense. It is about chemical access, thermal tolerance, stable attachment, microbial partners, and the constant negotiation between food and harm. A black smoker chimney can grow several meters tall, but it begins from small mineral deposition around an opening. Fluid finds a path. Minerals fall out of solution. A wall forms. The wall focuses flow, which encourages more deposition. Over time, the structure can become a tower with internal channels. Animals settle on the outside, but the inside remains a plumbing system. If the flow becomes blocked, pressure can force fluid elsewhere. If the chimney wall weakens, it can break. If new cracks open in the seafloor, the center of activity can migrate. A mature colony may therefore be living beside a system that is already changing beneath it. The architecture of the vent is temporary in geological terms, but it is long enough for animals to make it home. This mismatch between planetary time and animal time defines much of the deep sea. What is brief for rock can be a lifetime for a worm. The ROV's navigation track across a vent field can look messy after the dive, lines curving around chimneys, loops where the pilots circled a sample site, pauses where scientists argued over what they were seeing, a sudden retreat when the plume blinded the cameras. That track is part of the evidence too. It shows how hard it is to observe a three-dimensional, chemically active habitat from a vehicle that must avoid collision, protect its tether, maintain position, and keep delicate instruments alive. Some parts of the field may remain unseen simply because the terrain was dangerous for the machine. Some animals may be missed because they hid in cracks, under shells, or inside the subsurface. Some behaviors may stop when lights and vibrations arrive. Deep-sea science is often built from incomplete coverage. The responsible response is not despair. It is careful inference, repeated observation, and clear separation between what was seen, what is likely, and what remains unknown. Around underwater volcanoes, the scale of pressure is easy to state and difficult to imagine. Every square centimeter of an animal's body is under compression from the water above. Yet the animal does not feel pressure as crushing in the human sense because its internal fluids and tissues are adapted to the same pressure. The danger comes when it is removed from that world too quickly, or when surface-adapted organisms try to enter. Pressure affects membranes, proteins, gas spaces, and chemical equilibria. Deep-sea animals often lack compressible spaces like lungs, and their biochemistry functions under conditions that would disrupt shallow organisms. The ROV descends with housings built to resist what the animals simply inhabit. The machine fights the deep. The animals belong to it. That difference is one reason they can seem alien even when their ancestry is entirely earthly. A hydrothermal vent may be discovered by a chain of indirect signs. Multi-beam sonar maps the seafloor and reveals ridges, volcanic terrain, or mounds. Water column sensors detect chemical anomalies or particles. Towed cameras show mineral deposits. An autonomous vehicle follows a plume. Finally, an ROV or submersible reaches the bottom and records the active source. Each step narrows the search. Each step also has uncertainty. A plume can drift from its source. A sensor can detect a signal without revealing the chimney. A map can show terrain without life. The moment the vent appears on camera feels sudden, but it is usually the result of patient inference. The chimney enters the beam because people followed clues through dark water, from chemistry to geology to image. That process mirrors the habitat itself, hidden reactions becoming visible only where the right tools meet the right flow. The deep sea is often called poorly explored, and that is true in a practical sense, but it should not be turned into the claim that humans know nothing. Scientists have mapped many ridges, described many vent species, analyzed microbial pathways, measured fluid chemistry, and returned to long-term study sites. At the same time, vast areas remain unmapped at high resolution, many vent fields are undiscovered, and many organisms have not been observed through their full life cycles. The honest position is between ignorance and certainty. We know enough to explain why vent communities exist. We do not know enough to predict every field, every connection, every response to disturbance, or every hidden habitat beneath the crust. The deep ocean is not blank. It is partially read. The unread parts are not empty pages, but places where instruments have not yet been, or have not stayed long enough. The animal that seems most alien may depend on what the camera happens to show. A tube worm without a mouth challenges basic expectations of animal feeding. A shrimp swarm looks like a living blizzard against black rock. A Pompeii worm suggests an animal pressed against heat at the edge of tolerance. A scaly foot snail seems to carry a piece of the vent's mineral world on its body. A pale crab walking over red plumes gives the scene a familiar predator moving through unfamiliar architecture. Each one represents a different solution. Internal symbiosis, external microbial farming, grazing, scavenging, armor, heat tolerance, chemical positioning. None of these solutions is universal. The vent community is not strange, because every animal has the same impossible adaptation. It is strange because many unrelated animals have found different ways to use the same rare source of energy. The risk of calling vent animals alien is that it can make them seem separate from care. If something feels too strange, too remote, or too unlike us, it can become easy to treat as expendable. But these animals have ranges, life histories, vulnerabilities, and ecological roles. They are not ornaments on mineral deposits. They are the living expression of a habitat that may be rare, ancient in process, and fragile in particular locations. Deep sea mining, climate-driven ocean change, pollution, and expanding industrial activity all press against the old assumption that depth alone protects life. The deep ocean is no longer unreachable in the practical sense. Machines can descend. Claims can be made. Samples can be taken. The question is whether understanding arrives before damage, and whether the strange animals around underwater volcanoes are treated as communities to be studied with restraint rather than obstacles sitting on useful rock. A vent field in darkness is also a reminder that life does not require comfort. It requires usable energy, chemistry that can be harnessed, structures that can be occupied, and lineages capable of adapting to the available conditions. Comfort is a human projection. For a tube worm, the right place is not mild. It is a flow where sulfide and oxygen can both be captured. For a shrimp, the right place may be a turbulent chimney face where bacterial growth can be maintained. For a Pompeii worm, the right place may be a tube on hot mineral rock with cooler water near the head. These are not easy places. They are suitable places. The distinction matters. Life around underwater volcanoes is not proof that life can live anywhere. It is proof that life can become extremely precise when evolution has enough time, variation, and opportunity. Vent ecosystems also force a different way of thinking about productivity. At the surface, a bloom of phytoplankton may spread across huge areas visible from satellites. A vent bloom is hidden, local, and vertical, built around cracks and chimneys. Its productivity may be high within meters of flow, then drop sharply into the surrounding deep sea. From above, nothing announces it. No color spreads across the sea surface. No birds circle over it. A ship must know where to look, or must search for chemical traces. The world's surface can remain calm while a dense animal community lives below, powered by fluids that began as seawater sinking into rock. This hiddenness is part of the vent's identity. The ecosystem is not small because it is unimportant. It is small because the condition that feeds it is concentrated, and hidden because the ocean above it is deep enough to erase every ordinary sign. The relationship between volcanic activity and life is not always local or gentle. Large submarine eruptions can affect water chemistry, create plumes, bury habitats, and reshape seafloor terrain. Smaller events can redirect flow or open new hydrothermal paths. Even without eruption, tectonic movement can fracture rock and change circulation. Vents are expressions of a living planet in the geological sense: heat moving outward, plates shifting, crust forming and breaking, seawater reacting with minerals. The animals live on the biological edge of those processes. A mussel bed does not experience plate tectonics as a concept, but its existence may depend on a spreading ridge. A worm tube does not know the mantle, but its food web depends on heat that began far below. This is one of the reasons vent life feels larger than its physical footprint. It turns planetary motion into animal tissue. Some vent fields contain animals that are newly described or still being studied, and even well-known species can have unresolved life histories. How long do individuals live under different flow conditions? How often do larvae successfully settle? Which cues matter most? How do symbiont communities change across development? How do predators affect community structure? How resilient is a field after disturbance? The answers vary, and for many organisms they remain incomplete. Deep sea biology often advances by combining limited observations with careful sampling and models, then revising those models as new dives reveal more. This uncertainty should not be smoothed away in narration. It is part of the habitat. The deep ocean does not give itself all at once. Around vents, even a crowded frame can be scientifically incomplete, because the frame shows bodies but not every process that made them gather there. The light from the vehicle often turns drifting mineral particles into a kind of weather. They move past the lens, bright for a second, then vanish. Some may have formed moments earlier, when vent fluid met seawater. Some may be older materials stirred from the chimney. Some may be biological fragments, mucus, bacterial clumps, or sediment lifted by the vehicle. In deep sea footage, particles are not visual noise alone. They can indicate flow direction, turbulence, organic matter, mineral precipitation, or disturbance. They show that the water is active, even when the seafloor appears still. A shrimp moving through that particle field is moving through chemistry made visible. A tube worm plume extending into it is sampling a constantly changing mixture. The beauty of the scene comes from the fact that the haze is not decoration. It is part of how the habitat works. Around some vents, bacterial mats are grazed heavily by limpets and other small animals. Grazing can keep surfaces open, shape microbial community structure, and influence where larvae settle. Predators then feed on grazers. Larger scavengers feed on dead animals. Shells and tubes create three-dimensional structure that shelters juveniles and smaller species. A vent field can therefore build complexity from a simple energetic beginning. Chemicals feed microbes. Microbes feed hosts or grazers. Hosts create habitat. Habitat supports other animals. Animals die and feed scavengers and decomposers. Each layer adds texture. The result is not a miracle outside ecology. It is ecology under unusual rules. The same broad processes seen in forests, reefs, and seagrass beds, competition, facilitation, predation, succession, disturbance, are present, but they occur in darkness around hot mineral water instead of sunlight.
A surface viewer may imagine the deep sea as silent because human senses fail there. But many vent animals likely use chemical and tactile information more than vision. Shrimp antennae, crab legs, worm plumes, mussel gills, and microbial surfaces all participate in sensing or exchange. Chemical cues may guide larvae. Flow direction can indicate where food or danger lies. Contact with surfaces matters in a crowded chimney community. We should be cautious about imagining these sensory worlds too vividly, because direct evidence is limited for many species. But it is clear that the vent environment is full of information that a human eye cannot read. The darkness is not empty to the animals. It is structured by current, temperature, sulfide, oxygen, touch, vibration, and the presence of other bodies. The ROV adds vision for us. It does not define what the habitat means to them. When scientists bring back a chimney sample, it may continue to change in the container. Minerals oxidize. Microbes respond to new temperature and pressure. Fluids mix with seawater in ways they did not at depth. A sample that seemed stable on the seafloor begins to record the disturbance of being removed. This is why some instruments try to capture fluids in sealed bottles, maintain pressure, or measure chemistry directly at the source. Hydrothermal fluid chemistry can include sulfide, methane, hydrogen, metals, and other compounds, but concentrations vary widely between sites. The chemistry depends on the rock, heat source, circulation path, and mixing with seawater. No single bottle represents all vents. No single vent represents all underwater volcanoes. Every sample is a point in a wider pattern, valuable because it is specific, not because it explains everything. The deep ocean has many ways of making animals look otherworldly. Pressure makes bodies soft and gelatinous in the midwater. Darkness favors bioluminescence, large eyes in some species, reduced eyes in others, and long sensory appendages. Food scarcity favors patience, low metabolism, opportunism, and efficient feeding. Vents add another layer: chemical energy and heat from the crust. This is why the animals around underwater volcanoes should not be treated as merely strange-looking. Their strangeness is functional. A tube worm plume is not an ornament. A shrimp swarm is not just spectacle. A bacterial mat is not background. A mineralized snail covering is not fantasy armor. Each feature belongs to a set of pressures and opportunities. The deep sea shapes bodies slowly, but vents can shape communities quickly by concentrating energy in a place where most of the surrounding world has very little. The more humans learn about these ecosystems, the more obvious it becomes that discovery is not separate from responsibility. Naming a species can make it visible to science. Mapping a vent can make it visible to industry. Filming a community can inspire protection or curiosity, but it can also turn a place into a target. This tension is not unique to vents, but vents make it sharp because their mineral deposits and biological communities occupy the same physical structures. The chimney that holds copper and zinc may also hold worms, snails, shrimp, microbes, and the chemical flow that sustains them. Deep-sea exploration cannot pretend to be innocent simply because the water is deep. Every descent now happens in a world where human reach is expanding. The question is whether knowledge of alien life around underwater volcanoes will make the habitat seem more valuable alive than broken.
A vent field can be beautiful, but beauty is not the reason it matters. It matters because it expands the known range of life's energy systems, because it connects geology and biology directly, because it supports species with unusual adaptations, because it reveals how ecosystems assemble under extreme conditions, and because it offers analogs for thinking carefully about habitable environments beyond Earth. The footage is simply the doorway. The black plume, the white tubes, the shrimp, the crabs, the mineral towers, the shimmering water all invite attention. Then, the science deepens the image. It asks where the carbon comes from, how the bacteria fix it, how larvae find the habitat, how the chimney grows, how long the flow lasts, and what happens when the volcano changes. The scene becomes more astonishing when it is explained, not less. A failing vent does not end in a single cinematic collapse. Often, it fades. Flow weakens, temperatures shift, bacterial mats thin. Symbiotic animals lose access to the chemistry they need. Mobile animals adjust their positions. Sessile animals remain where they are. Predators and scavengers respond to stress and death. The mineral structure persists after the living abundance declines, like a building after its power has been cut. Over time, inactive sulfide deposits may become habitat for other deep-sea organisms that use hard substrate rather than chemical energy. Corals, sponges, or other animals may colonize depending on location and conditions. The volcano's gift is not permanent in one place. Life follows the active flow while it can. Then other forms of life may inherit the structure. The same chimney can be a hot chemical engine, a dying colony, and later a cold rock surface in the dark. The idea of succession around vents has a practical importance. If a site is disturbed, recovery depends on whether nearby populations can supply larvae, whether suitable flow remains, whether the physical substrate is intact, and whether the chemistry still supports the same community. A destroyed chimney cannot be recolonized in the same way if the hydrothermal plumbing is gone. A buried field may not recover if active flow does not return. Natural eruptions and human disturbance are not identical, even if both can damage communities. Natural disturbance may create new habitat as it destroys old. Human disturbance may remove structures without creating the right new conditions. This is why arguments about resilience must be specific. Vent life can recover from some natural disturbances, but that does not mean any disturbance is harmless. The ability to colonize new cracks is not a guarantee against losing a habitat faster than it can be replaced. Sometimes the camera finds the edge of the vent field, and the scene changes quickly. The tubes stop, the mussels thin, the rock becomes darker and less crowded. A few animals remain, then fewer. Sediment appears in pockets. The plume is behind the vehicle now, and the water ahead looks like ordinary deep sea again. That transition may happen over tens of meters. It shows how concentrated the energy source is.
It also shows how dependent the animals are on the chemistry of the flow. The surrounding seafloor is not lifeless, but it is different. It runs on a thinner supply of food. Its animals are spaced differently. Its rhythms are less tied to hot fluid rising from the crust. The edge of a vent is one of the clearest boundaries in the deep ocean, not because a wall exists, but because energy fades.
A vent community can also expand upward into the water through the behavior of animals. Shrimp can swim or swarm above the chimney, fish can move in and out, larvae enter the plankton, plume microbes rise above the source, particles settle elsewhere. Predators may carry vent-derived carbon into nearby habitats. If scientists analyze tissues using isotopes, they can sometimes distinguish carbon sources and trace how much of an animal's diet comes from chemosynthetic production versus photosynthetic material from above. These chemical fingerprints help reveal connections that the camera cannot follow. A fish seen at the edge of the vent might be a visitor or a resident. A crab may feed on vent animals but move across non-vent ground. A small invertebrate may depend on microbial films that only form in diffuse flow. The food web is not just who eats whom. It is where the carbon came from before any mouth or claw touched it. The language of extremes can make vent animals sound as if they seek maximum heat or maximum toxicity, but most live in moderated conditions near mixing zones. The hottest fluid is usually too extreme for animal tissues. The cold background water lacks the reduced chemicals needed by many symbionts. The usable habitat lies between those endpoints. This distinction matters for accuracy. A tube worm is not bathing in 400-degree water. A shrimp swarm is not living inside the central black plume. A Pompeii worm may occupy a hot chimney surface, but different parts of its body experience different temperatures. The drama is not that animals ignore limits. The drama is that they live so close to limits and use them. Their bodies are positioned where chemistry becomes food before it becomes fatal, and where cold water tempers heat without erasing the chemical signal. The seafloor around a vent can look industrial, almost manufactured, because sulfide chimneys resemble pipes, towers, furnaces, or broken machinery. This resemblance is deceptive. No one built the structures. They assembled from reactions between hot fluid and cold seawater, from precipitation, collapse, and renewed flow. The animals did not move into a ruin. They moved onto active geology. A chimney wall may contain iron, copper, zinc, and sulfur minerals. Its outer surfaces may support microbes. Its cracks may leak warm fluid. Its base may be littered with fallen fragments and shells. When the ROV lights sweep across it, the scene can feel like entering a city, but it is better understood as a reef of chemistry, a mineral construction whose architects are water, heat, pressure, and time. The animals are tenants only as long as the plumbing continues. Deep time enters the vent story differently than it enters a fossil story. Individual vents may be short-lived, but hydrothermal circulation has likely occurred along ocean ridges for as long as plate tectonics and seawater have allowed it. The species are modern. The process is ancient. This distinction prevents exaggeration. A living tube worm is not a fossil from early Earth, and a modern vent field is not unchanged since life began. But the physical relationship between seawater, hot rock, chemical gradients, and microbial metabolism may echo very old planetary processes. Around underwater volcanoes, time is layered. A shrimp lives for its own span. A chimney grows over years or decades. A ridge opens over millions of years. The ocean crust is created and eventually destroyed in subduction zones. The process that feeds the animals is temporary in place and persistent in principle. The return from a dive can feel abrupt. The vehicle leaves the chimney, rises into black water, and the seafloor disappears. For a while, particles move through the beam. Then there is only water and instrument data. The ROV passes through the midnight zone, then the twilight zone, then into blue light, and eventually the surface where waves slap against the ship and the air seems ordinary. Samples come on deck in boxes and bottles. Hard drives fill with footage. Scientists begin the slower work of logging, measuring, sequencing, comparing, and questioning what they saw. The vent remains below, still flowing or changing without the camera. Animals continue feeding, withdrawing, settling, dying, and being eaten. Human attention comes into sense. The ecosystem continues between them. This is one of the quiet humilities of deep-sea research. The most important parts of the animal's life happen when nobody is watching. The title promises alien life, and the temptation is to end by making the vents feel unknowable. They are not unknowable. They have been sampled, mapped, measured, filmed, modeled, and studied by generations of scientists. Their microbes can be sequenced. Their fluids can be analyzed. Their animals can be described. Their distributions can be mapped against gradients. What remains is not pure mystery, but unfinished knowledge. That is a more durable kind of wonder. We know why animals gather around underwater volcanoes. We know chemistry can replace sunlight as the first step in a food web. We know symbiosis can build large bodies from microbial work. We know vents can be disturbed, recover, shift, and disappear. We also know that new cavities, species, behaviors, and connections are still being found. The deep sea keeps adding rooms to a house science has already entered. The camera light returns to the chimney, and the scene is again physical. White tubes stand against black rock. Shrimp gather near a wavering flow. A crab steps over a fallen shell. Mineral particles rise into the dark water and vanish above the frame. Nothing here is outside nature. Nothing here is supernatural. The animals are not proof that life has no limits. They are proof that limits can become habitats when chemistry, evolution, and time meet in the right way. The underwater volcano does not glow like a storybook furnace. It leaks heat through cracks. It stains seawater with minerals. It builds towers and breaks them. Around those towers, animals make a living from reactions that begin in rock. The rove pulls back. The plume folds into darkness, and for a few seconds the red plumes of the tube worms remain visible. Then, the beam moves on, and the vent continues without us. Before the vehicle ever reaches a vent, it passes through a long descent that makes the surface seem increasingly unlikely. The first 100 meters still belong to ordinary ocean color, with blue light and drifting plankton and the memory of waves. Below that, the water begins to lose the parts of sunlight that human eyes use most easily. Red is gone. Orange is gone. Green fades. The last blue dims until it becomes a trace instead of illumination. In the mesopelagic zone, animals still respond to the surface day. Lanternfish, krill, squid, and gelatinous predators move through a world where light is measured in faint gradients rather than bright scenes. Many migrate upward at night and descend by day, carrying carbon through the water column in their bodies. The vent is still far below them. It does not participate in sunrise. But the descent through these layers matters because it reminds us that the deep sea is not a single darkness. It is a stack of habitats, and the volcanic seafloor is one end of a long vertical story. By the time the rove reaches the abyssal or bathyal seafloor, depending on the site, the surface has become an abstraction expressed mostly through oxygen and marine snow. Small particles fall from above, some fresh, some degraded, some eaten and repackaged many times before they arrive. Animals on ordinary deep sediment depend on this slow delivery. Sea cucumbers process mud. Brittle stars lift arms into faint currents. Worms burrow through sediment. Microbes break down what remains. Then, the vehicle crosses into a hydrothermal field, and the logic changes. Food is not only arriving from above, it is being made locally from chemical energy. The camera does not show that first step directly, but it shows the result in density. A place that should have looked like cold rock becomes crowded. The contrast is one of the most important images in deep-sea science. Scarcity around the field, abundance at the crack, both real, both part of the same ocean. The oceanic crust beneath many vents is young compared with continental rocks, and it is constantly being made at spreading centers. Magma rises, cools, cracks, and becomes basalt. Seawater enters those cracks. Heat drives circulation. The water reacts with rock and returns changed. It is a planetary-scale plumbing system, but locally it may reveal itself through a hole small enough for an animal to cover. The pressure forces everything into close contact. Water is pushed through fractures. Minerals are carried and deposited. Microbes colonize surfaces where energy becomes available. Animals gather where the microbial production can be reached. This means a vent community is not only living near geology, it is living inside a geological process that has become biologically useful. The crust is not a foundation in the passive sense. It is a reactor, a filter, a heat exchanger, and a hidden reservoir of chemical possibility. The term chemosynthesis can sound like a single process, but it includes many pathways and organisms. Some microbes oxidize sulfide. Some use hydrogen. Some use methane. Some processes require oxygen, while others may use nitrate or other electron acceptors depending on conditions. The details change from site to site and even across centimeters within one microbial mat. This diversity matters because animals at vents do not all rely on the same chemistry. A mussel with sulfur-oxidizing symbionts is not identical to one with methane-oxidizing partners. A shrimp carrying microbial communities on its mouthparts or gill chamber is not the same arrangement as a tubeworm housing bacteria deep in a trophosome. The word chemosynthesis is a doorway, not the whole room. Once inside, the vent becomes a network of specific reactions, each tied to a living strategy and each dependent on the chemistry moving through the rock. A black smoker can look hostile enough that the animals nearby seem misplaced, but their location is exact. The central plume may be too hot and chemically intense, while the surrounding seawater is too cold and chemically poor for the same kind of production. At the boundary, turbulent mixing creates usable conditions. This is where tubes cluster, where shrimp gather, where microbial mats thicken, where animals feed through partners, or by grazing on those partners. The animals are not drawn to danger for its own sake. They are drawn to an energy boundary. The danger is part of the cost. If the boundary moves suddenly, bodies can be trapped in the wrong water. A change in flow can expose tissue to heat or remove the sulfide supply. A plume that fed a colony yesterday can starve it tomorrow. Life at vents is abundant because energy is abundant, but abundance does not mean stability. Tubeworms provide one of the clearest examples of how anatomy can be reorganized around symbiosis. The animal's plume collects gases and dissolved compounds from the surrounding water. Its circulatory system transports them. Its trophosome houses bacteria that use chemical energy to fix carbon. The worm's body receives nutrition from those bacteria. The tube protects the soft animal and anchors it to the seafloor. Everything about the adult form points toward this arrangement. The missing digestive tract is not a deficiency, it is part of a different design, one that would make no sense without the vent. If a tubeworm were described without its habitat, it would seem incomplete. In place, it is complete in a way that depends on chemistry outside itself. This is true of many vent animals. Their bodies are not fully understandable until the water around them is included. A mussel bed near a vent can be thick enough to form a living pavement. Shells overlap. Small animals move through crevices. Sediment and mineral grains collect in the spaces between them. The mussels may host symbiotic bacteria in their gills, using chemical energy while also interacting with particles in the water. Their shells provide hard surfaces in a habitat where surfaces are contested. Juvenile organisms settle among them. Predators search the bed. Dead shells remain as structure after the animals inside are gone. In the camera lights, the bed may look less dramatic than a black smoker, but it is no less a constructed habitat. The animals are building complexity by being present. A small worm living between two mussel shells is not living beside the vent in a broad sense. It is living in a space created by other animals that depended on the vent before it did. Shrimp swarms can be difficult to count because the bodies overlap until the chimney seems to have become a moving surface. Some cling to rock, some crawl over one another, some swim briefly and settle again. The group responds to flow, disturbance, light, and perhaps heat or chemical cues. Rimicaris exoculata and related vent shrimp are among the best-known examples in Atlantic vent systems, and studies have shown close associations with microbial communities that help them exploit chemical energy. Their eyes are reduced in ordinary visual terms, but specialized organs may detect cues near the vents. Calling them blind is convenient, but it can hide the fact that their sensory world is not simply a poorer version of ours. It is tuned to a habitat where vision in the human sense is not the main guide. Their bodies read a chimney through contact, chemistry, heat, and collective position. The Pompeii worm's fame comes from heat, but heat alone does not explain it. Its tube, microbial covering, circulation, behavior, and the structure of the chimney all matter. The animal is often positioned across strong temperature gradients, with one part of its body near a warm fluid and another closer to cooler seawater. Measurements near these animals can vary sharply because the environment is turbulent and patchy. This is why careful researchers avoid treating a single high number as if it were the animal's whole experience. The worm lives at a boundary, not in a uniform furnace. That boundary is still extraordinary. A soft-bodied annelid, related broadly to worms found in many marine environments, has become capable of occupying mineral chimneys where the surrounding conditions would exclude almost every familiar animal. Its strangeness is not that it ignores biology, it is that biology has found a narrow way through. On inactive parts of a vent field, the remains of earlier life can look like abandoned scaffolding. Empty tubes stand without red plumes, shells lie in piles. Mineral crust covers surfaces that once leaked warm fluid. A crab may still cross the area, but the dense feeding activity has shifted elsewhere. These remains are valuable evidence. They show where flow used to be. They preserve the outline of former communities. They can be dated, measured, and compared with current activity. Death at a vent is not a separate chapter from life. It is part of the map. The distribution of empty tubes, broken chimneys, and shell beds tells scientists how the field has changed. Around underwater volcanoes, the past is not always buried deeply. Sometimes it stands in the camera beam as a white tube that no longer holds a worm. A newly opened vent is a different scene. Fresh basalt may be dark and glassy. Fluid may shimmer through cracks before a large chimney has formed. Bacterial films may be the first clear sign that chemistry is entering biology. Small grazers may appear before larger animals dominate. If larvae arrive and conditions remain favorable, the field can begin to assemble into a recognizable community. The early stages are important because they show how life responds to opportunity. Animals do not simply wait in the rock, fully formed. They arrive, settle, fail, survive, grow, and compete. A mature tubeworm colony can make the process look inevitable, as if the vent was always destined to be covered in white tubes. The younger field tells a more uncertain truth. It could become a dense community, or the flow could fade before the community fully forms. The ocean connects vents through movement that is hard to see. Deep currents can carry larvae along ridges, across fracture zones, or into regions where they will never find suitable habitat. Hydrothermal plumes can rise and spread, creating chemical traces that may influence dispersal. Topography can trap water or steer it. Yet connection is not guaranteed. Some vent species show genetic separation between regions, suggesting barriers or limited exchange. Others appear more connected across distances. The pattern depends on larval biology, current structure, habitat spacing, and history. A map of vent fields can look like a series of points, but for an animal larva, those points are not equal. Some are reachable. Some are effectively distant beyond survival. Some may exist undiscovered. The deep ocean is continuous as water, but discontinuous as habitat. Many vent animals are endemic, meaning they are found only in vent habitats or even in particular vent regions. Endemism is common in environments that are isolated, specialized, or patchy. It raises the stakes for disturbance because losing a habitat can mean harming species with limited alternative homes. This does not mean every species at every vent is restricted to one chimney. It means that the geography of vent life must be treated carefully. A species may be common at one ridge segment and absent elsewhere. Another may be widespread across connected fields. Another may be known from so few sites that its real range is uncertain. The deep sea teaches humility here. Absence in samples may mean true absence, or it may mean we have not looked in the right place. Conservation has to work with uncertainty without pretending uncertainty is permission to ignore risk. The machinery used to study vents is shaped by the same extremes that shape the animals. Cameras need housings that resist pressure. Lights must work in cold water. Thrusters must hold position in turbulence. Temperature probes must survive steep gradients. Fluid samplers must capture hot, reactive water without losing the chemistry that made it important. Robotic arms must be strong enough to break rock and delicate enough not to destroy the sample. A research vessel above may look like a collection of cranes, cables, containers, screens, and wet decks. Beneath it, the ROV becomes the hands and eyes of the expedition. This human technology does not make the deep sea familiar. It makes temporary contact possible. The machines are impressive, but their most important role is modest: to hold still in a place where humans cannot stand and let the animals reveal a little of how they live. Sometimes the most revealing moment in vent footage is not the black smoker itself, but an animal crossing between zones. A crab leaves a shell bed and enters a warm patch. A fish moves toward the shrimp and then turns away. A tubeworm plume withdraws when touched by a current shift or disturbance. A shrimp swarm opens briefly around a hotter flow, then closes again. These movements show that the animals are actively managing conditions. They are not simply stuck to the vent like mineral deposits. Even sessile animals can regulate exposure through plumes, tubes, and internal physiology. Mobile animals can choose positions, retreat, or exploit short-lived opportunities. The vent field is therefore not only a set of zones drawn by chemistry, it is a place of behavior where animals respond continuously to a habitat that changes around them in seconds, seasons, eruptions, and geological intervals. Reproduction at vents is still one of the most difficult subjects to observe directly. Many adult animals are visible, but spawning events, fertilization, larval development, settlement, and early growth often happen beyond the camera's schedule. Scientists infer reproductive strategies from anatomy, larval collections, genetics, laboratory work, and repeated surveys of juvenile stages. Some vent species release eggs and sperm into the water. Some brood young. Some larvae feed. Others rely on yolk. Some may delay development under cold deep-sea conditions. The range of strategies is broad because vent animals belong to many lineages. This matters because the persistence of a vent community depends not only on adults surviving the present flow, but on offspring finding the next suitable place. Around underwater volcanoes, reproduction is a dispersal problem, a timing problem, and a chemical navigation problem all at once. The deep sea has no shortage of hard surfaces, but the right hard surface is rare. Fresh basalt, chimney walls, inactive sulfide, sedimented lava, carbonate towers, and old shells all offer different settlement conditions. Microbial films change those surfaces. Mineral chemistry changes them. Flow changes them. A larva ready to settle may need cues that indicate not just something solid, but something alive with the right chemical possibility. It may be that a surface smells right in a chemical sense, or that microbial biofilms provide signals, or that temperature and flow combine into a cue. The details vary and are still studied. From a human view, settlement may look like a tiny animal attaching to rock. From the animal's life history, it is the decisive moment, the point where a drifting body becomes part of a vent field or fails to become anything at all. The animals around vents have enemies beyond predators. Flow can stop. Lava can arrive. Sediment can smother. Toxic concentrations can rise. A chimney can fall. A parasite or disease can spread through dense clusters. Human equipment can disturb. Mining could remove substrate. Climate change can alter the water masses that supply oxygen and background chemistry. Not all of these threats operate everywhere or with the same force. The point is not to make the vent sound doomed. It is to make it real. Real habitats have multiple pressures. Real animals have tolerances. Real communities can be resilient in some ways and vulnerable in others. The deep sea is often protected in imagination by distance, but distance is not a shield once technology reaches it. A habitat hidden for most of human history can become accessible faster than it becomes understood. Hydrothermal vent research has also changed microbiology beyond the deep sea. Enzymes from heat-loving microbes have helped scientists understand molecular stability under extreme conditions. Studies of vent archaea and bacteria have expanded knowledge of metabolic diversity. The discovery of thriving communities without sunlight changed how biology classes, astrobiology programs, and oceanography textbooks describe the range of life. Yet it is worth remembering that vents are not valuable only because they may help humans. They are valuable as ecosystems in their own right. Their animals do not exist to provide analogies, enzymes, minerals, or inspiration. A tubeworm colony matters first because it is a living community, because it is part of Earth's biodiversity, because it represents a way of being alive that evolved under conditions humans barely enter. The uses we find for that knowledge come after the existence of the place itself. The microbial world beneath and within vent communities may be more continuous than the animal world above. Microbes can occupy pore spaces, chimney interiors, plume water, animal tissues, and diffuse flow surfaces. They can respond quickly to changes in chemistry and temperature. Some can tolerate conditions that exclude animals entirely. This means there may be microbial habitats where the camera sees no animals, and animal habitats that depend on microbial activity in places the camera cannot reach. The visible community is only the upper layer of a much more complicated biological system. Scientists are still working to understand how deep the biosphere extends below vents, how energy limits microbial communities in crustal fluids, and how those subsurface microbes interact with animals above. The more the seafloor is sampled, the less the boundary between ocean and crust looks like a simple line. If a pilot turns off the ROV lights for a moment, the vent disappears to human sight. The plume, animals, rocks, and tubes remain, but the camera becomes blind. Some animals may sense the change only as the removal of an artificial disturbance. Others may continue exactly as before. Bioluminescence is common in many deep-sea animals, especially in the water column, but vent communities are not primarily organized around light. Chemical and physical gradients matter more. There may be faint thermal glow from very hot vents under certain conditions, and some shrimp sensory organs may detect aspects of vent radiation, but the habitat is not lit in the way footage suggests. The darkness is natural there. The lights are the event. When the lights go out, the animals are not lost. We are. The edge of a brine pool, the bones of a whale fall, the seepage of methane from sediment, and the hot flow of a vent all show different ways the deep sea concentrates energy. They are not the same habitat, but they belong in the same broader lesson. The deep ocean can look empty until energy gathers in a place. And then, animals find it. Whale falls feed scavengers and bone-eating worms through organic remains. Cold seeps support chemosynthetic communities through methane and sulfide. Brine pools create sharp chemical boundaries that some animals approach and others avoid. Hydrothermal vents use heat-driven circulation through volcanic rock. The deep sea is therefore not a uniform desert. It is a landscape of patches, some sparse, some crowded, some temporary, some long-lived, each structured by the form energy takes when it arrives. Around vents, energy arrives as disequilibrium. Hot fluids rich in reduced chemicals meet cold oxygenated seawater. Life exploits the fact that those chemicals have not yet reached equilibrium. Microbes capture energy as reactions proceed, and some of that energy becomes biomass. This is a profound but concrete idea. The animals are not living from heat directly in the way a machine might use steam. They are living from chemical reactions made possible by heat, rock, water, and mixing. This is why not every warm place in the deep sea supports the same kind of life. The right chemicals must be present, and they must mix with the right oxidants under conditions organisms can tolerate. Heat alone does not feed a tubeworm. Chemistry in motion does. The comparison with surface volcanoes can mislead in another way. On land, volcanic landscapes often begin as bare rock and ash exposed to sunlight, rain, wind, and colonization by lichens, plants, insects, and eventually more complex communities. Underwater, fresh volcanic rock enters a cold, dark, pressurized ocean. If hydrothermal circulation develops, microbes may colonize chemically active surfaces quickly. Animals arrive through larvae or movement rather than seeds or flying insects. Succession still happens, but its materials are different. There is no grass, no shrub stage, no forest canopy. There are microbial mats, tubes, shells, shrimp, snails, crabs, fish, and mineral surfaces. The underwater volcano becomes green in no ordinary sense. It becomes alive through white tubes, red plumes, pale bodies, and invisible carbon fixation in darkness. Vent science also contains mistakes, revisions, and changing interpretations, as all science does. Early excitement sometimes produced broad claims that later work made more precise. Temperature tolerances were refined. Species ranges changed as new vents were discovered. Relationships between animals and symbionts became more complex than first descriptions suggested. The role of subsurface habitats expanded after new observations. This does not weaken the field. It is how the field becomes reliable. The first view of an alien-looking community is powerful, but the second, 10th, and 100th view are what turn astonishment into understanding. A good narration should leave room for that process. The goal is not to freeze vents as a single shocking discovery from 1977, but to show a living area of research where each dive adds detail to a habitat that was already extraordinary before it was famous. The animals at vents often appear packed tightly enough that competition for space must matter intensely. Tubeworms need access to flow. Mussels need attachment sites and exchange with the water. Microbial mats occupy surfaces. Grazers remove films. Predators and scavengers move through the structure. A chimney face can become crowded because only some parts provide the right conditions. Space at a vent is not simply room. It is chemistry plus room. A bare patch of rock, away from flow, may be physically available but ecologically useless to a symbiotic animal. A crack with warm fluid may be valuable enough for many organisms to crowd around it. This changes how abundance should be understood. The animals are not crowded because the deep sea suddenly became generous everywhere. They are crowded because usable habitat is concentrated, and every body is responding to that concentration. Some animals at vents likely experience thermal and chemical stress routinely, not as rare disasters but as daily conditions. A plume wavers, flow pulses. A neighbor blocks access. A predator causes movement. The animal shifts or withdraws. Its tissues repair damage. Its microbes adjust. Stress is not always fatal. It can be part of the normal range of life in variable environments. But there is a limit beyond which adaptation fails. Understanding where those limits lie is difficult because laboratory conditions cannot perfectly recreate the vent. Researchers use experiments, in situ measurements, gene expression, physiological studies, and observation to estimate tolerances. The animals themselves give the most basic answer by their distribution. They show where life is possible by being there, and where it is not by leaving the rock bare. The red plume of a tube worm can vanish instantly into its tube when disturbed. This simple movement carries several meanings. It protects delicate tissue from predators or mechanical damage. It changes the worm's exposure to oxygen and sulfide. It makes the animal briefly disappear from the visible community. For a sessile animal, withdrawal is one of the few rapid behaviors available. The tube is not just a house, it is a defensive structure, a support, and a way to maintain position in flow. The camera may linger on the plume because it is colorful, but the tube is equally important. It marks the animal's commitment to a place. Once established, the worm's life depends on that spot remaining chemically suitable. The plume can withdraw from danger. The worm cannot move its entire future somewhere else. A shrimp swarm responds differently. It can part around the rove arm, flow over the chimney like a living surface, or scatter briefly before gathering again. Individual shrimp are mobile, but the swarm remains tied to the chimney because the food source is tied there. This creates a tension between movement and dependence. A shrimp can leave a harmful patch more easily than a tube worm can, but it still needs the microbial production associated with vent flow. It has freedom measured in centimeters or meters, not in oceans. The swarm's movement can make the habitat look more flexible than it is. The animals shift constantly, but the colony as a whole remains anchored to chemistry. If the vent dies, motion alone cannot save the system unless another suitable vent lies within reach. The carbon fixed at vents can be traced using stable isotope signatures because chemosynthetic pathways often leave different chemical patterns than photosynthetic production from the surface. Scientists analyze tissues to understand who is eating vent-derived carbon and how it moves through the community. These measurements help separate appearances from actual diet. A predator near a vent may rely heavily on vent animals, partly on surrounding deep-sea prey, or on a mixture. The body becomes a record of meals, and the meals become a record of energy source. This kind of evidence is less cinematic than a black smoker, but it is essential. It shows whether the vent is simply a place where an animal was seen, or a source of carbon that entered the animal's body. In deep-sea ecology, location and nutrition are not always the same question. There are also vents where the most important life may not be visually spectacular. A field with low animal density can still host diverse microbial communities and unusual chemistry. A diffuse flow area may appear as stained rock and pale mats rather than towering chimneys. A subsurface habitat may be invisible unless rocks are lifted. Human attention naturally follows large animals, but science cannot stop there. Microbial metabolism, fluid pathways, and mineral reactions may be the main story at some sites. This is another way the deep sea resists a simple documentary pattern. Not every important scene announces itself with a crowd of shrimp or a forest of tube worms. Some of the most revealing places are quiet on camera, and their significance emerges later in the lab when water chemistry, DNA, and mineral structure are compared. The vent field is also a place where boundaries between disciplines break down. A geologist sees basalt, sulfides, fluid pathways, and tectonic setting. A chemist sees redox reactions, dissolved metals, pH, gases, and precipitation. A microbiologist sees metabolisms, symbioses, and genetic diversity. A zoologist sees anatomy, behavior, reproduction, and community structure. An oceanographer sees currents, plumes, mixing, and water masses. The animals exist where all of these views meet. A tube worm cannot be understood by zoology alone, because its food comes from microbes using chemicals supplied by geology and mixed by ocean physics. This is why hydrothermal vents became such important places in science. They force the planet to be studied as a system, not as separate categories. Around underwater volcanoes, rock and water become biology without waiting for sunlight. The footage from vents often has a slow rhythm because the vehicle must move slowly. The camera approaches, steadies, adjusts focus, loses the plume in glare, backs away, tries again. A crab crosses the frame with no awareness of the patience required to film it. A shrimp antenna touches the lens housing. A worm plume withdraws. This pacing fits the habitat better than rapid spectacle. The important details are small. A shimmer in the water, a temperature reading, a pale film, juveniles on a fresh surface, a dead tube beside a live cluster. The viewer has to learn to read the scene almost the way scientists read it, not by waiting for a dramatic attack, but by noticing placement, density, flow, and change. The vent is most revealing when attention slows down enough for the ordinary lives of strange animals to become visible. A deep sea fish hovering near a vent may seem like a visitor from a more familiar ocean. It has eyes, fins, a mouth, a recognizable body plan. But its presence still raises questions. Is it feeding on vent shrimp or worms? Is it using warmth? Is it simply passing through a hard-bottom habitat where prey is abundant? How often does it enter the chemical flow? Does it reproduce nearby or only hunt there? For many fishes, direct observations remain limited. A few frames can document presence without explaining dependence. This is why scientists combine video with stomach contents, isotopes, genetics, and repeated sightings. The animal on screen is a clue, not a complete answer. In the deep sea, a familiar-looking fish can be just as scientifically uncertain as the most alien worm, because knowing what it looks like is not the same as knowing how it lives. The vent community has no trees, but it can have vertical structure. Chimneys rise above the seafloor. Tubeworm tubes stand upright. Mussels form layered beds. Shrimp stack on chimney walls. Plumes rise above the animals. Fish occupy water just off the bottom. Microbes coat surfaces from the base to the warmest cracks. This three-dimensional structure increases habitat complexity, creating shelter, feeding surfaces, flow variation, and attachment points. Small animals live in spaces created by larger ones. Predators navigate around towers and tube clusters. The result is not a forest in the botanical sense, but the comparison helps explain why a crowded vent field feels alive at several levels. It has a floor, walls, towers, canopies of plumes, and a water column carrying particles upward. In darkness, architecture can still organize life. Heat is not evenly distributed through that architecture. A chimney wall may be hot near an orifice and much cooler a short distance away. Diffuse flow may seep through cracks between animals. Warm water may pool briefly under overhangs before mixing away. This patchiness allows species with different tolerances and needs to occupy different positions. It also creates risk. A place that was suitable during one measurement may not remain suitable if flow shifts. Temperature maps of vent fields are therefore snapshots of a moving target. They are useful, but they must be interpreted with time and variability in mind. The animals do not live in the average temperature of a vent. They live in the changing temperature at the exact place their tissues meet the water. That precision is what makes the habitat both productive and precarious. The microbes in symbiosis with vent animals are not all located in the same places. Tubeworm bacteria live inside the trophosome. Some mussel symbionts live in gill cells. Some shrimp cultivate or carry microbial communities on body surfaces or in specialized chambers. Some snails and clams host bacteria in tissues. These arrangements show repeated evolutionary experiments with the same broad idea: let microbes do chemistry that animals cannot do alone, and build an animal life around the products of that chemistry. The partnerships are not interchangeable. A tubeworm cannot simply switch to a shrimp's method. A mussel's gill symbiosis is not the same as a worm's internal organ. Evolution did not discover one vent solution. It discovered many, separately, in different bodies, each shaped by ancestry as much as by environment. A living vent community can be compared to a market only if the comparison remains physical. Chemicals are supplied, microbes transform them. Animals provide housing, access, or surfaces. Predators and scavengers move biomass. Waste products return to the environment. Space is contested. Energy is lost as heat and metabolism. No organism understands the exchange, but the exchange structures the habitat. The worm cannot fix carbon without bacteria. The bacteria inside the worm cannot access sulfide and oxygen in the same way without the host. Grazers depend on microbial growth, which depends on flow. Predators depend on prey, which depend on microbes. The community works because many dependencies line up. It fails when enough of them are broken. Around underwater volcanoes, life is not independent of harshness. It is built by using harshness in controlled, negotiated forms. Some vent animals are damaged by the process of being seen. Bright lights may alter behavior. The rov's presence can change flow, stir sediment, or cause animals to move. Sampling removes individuals and structures. Even when researchers are careful, observation has a footprint. This does not mean exploration should stop. It means exploration should remain aware of itself. The deep sea cannot be studied without contact, but contact can be designed to minimize harm and maximize knowledge. Non-invasive imaging, long-term observatories, environmental DNA, careful sampling protocols, and protected areas can all help reduce unnecessary disturbance. The goal is not to pretend humans can be invisible. The goal is to enter briefly, learn honestly, and leave as much of the habitat functioning as possible after the vehicle returns to the surface. Environmental DNA has begun to change how scientists search for life in water and sediment. Organisms shed genetic material through cells, mucus, waste, eggs, sperm, and decay. By sampling water around a vent, researchers may detect species that were not seen on camera or were hidden in cracks. This method is powerful, but it has limits. DNA can drift. It can persist for a time after the organism has moved or died. It may not reveal abundance clearly without careful interpretation. It depends on reference databases that may be incomplete for deep-sea species. Still, it adds another layer to the evidence. The vent can now be read not only through images and specimens, but through traces dissolved in water. In a habitat where bodies hide behind chimneys and under lava plates, invisible evidence can reveal part of the crowd the camera missed. The discovery of animals beneath lava plates near vents feels surprising because people tend to imagine the seafloor as a surface. But volcanic seafloor can be fractured, layered, and full of cavities. Warm fluids can move through those spaces. Microbes can live there. Larvae may enter or be transported through them. Larger animals can occupy shallow voids where space and chemistry allow. The underside of the seafloor becomes another habitat, not deep in the crust in a vast cavernous sense, but in small cavities just below the visible world. It is important not to exaggerate this into a global underground animal kingdom. The finding is specific and still being investigated. But it changes the image of the vent. The community is no longer only what grows upward from the rock. It may also include life tucked underneath in the warm plumbing spaces below the camera's first view. When animals live below the visible seafloor, conservation and sampling become more complicated. A surface survey may underestimate habitat. Removing or crushing lava plates could affect animals not visible from above. Disturbance to flow paths could alter both surface and subsurface communities. This matters for any future activity that treats the seafloor as a two-dimensional mineral surface. Around vents, the habitat can have depth in a biological sense as well as a geological one. A map that marks chimneys and animal clusters is useful, but it may not capture the hidden cavities that support larvae or adults. The ocean floor is not simply the place where life sits. In some vent systems, it is part of the living space. Understanding that before disturbing it may be one of the most important lessons of recent research. A black smoker's plume can blind the ROV if the pilot moves too close. The screen fills with dark particles. Focus disappears. The chimney becomes a shadow. The pilot backs away, waits for the water to clear, and approaches from another angle. This small operational problem reflects a larger scientific one. The most active part of the vent is also one of the hardest to see and sample. Heat, particles, turbulence, and chemical reactivity interfere with observation. The plume is the source of much of the habitat's energy, yet it hides itself. Scientists must read it through temperature probes, fluid samplers, chemical analysis, and the distribution of animals around it. The camera beam can show the plume as a dark cloud, but the real information is inside the chemistry, moving too fast and too hot for a simple image to explain. It is easy to say that vents are independent of the sun, and in one narrow sense, the carbon fixation at vents does not require sunlight. But the oxidants used in many vent reactions, especially oxygen in deep seawater, are tied to the broader oxygenated ocean and atmosphere, which are products of photosynthesis over Earth's history and ongoing surface processes. This means vent ecosystems are chemically connected to the sunlit planet even when their local energy source is geological. The independence is real, but not absolute. Life at vents demonstrates that ecosystems can begin with chemical energy rather than sunlight, but the modern ocean is an integrated system. The deep water that bathes the vents carries the legacy of surface photosynthesis. The animals around the chimney live where Earth's interior chemistry meets water that has been shaped by the biosphere above. The first scientists who saw dense vent life were not simply finding new animals, they were finding a new kind of ecological foundation. Before vents, large animal communities in the deep sea were mostly explained by organic matter from above. After vents, chemosynthetic ecosystems became a major category, eventually including hydrothermal vents, cold seeps, whale falls in certain stages, and other reducing habitats. The discovery widened biology's imagination, but it did so through evidence, photographs, specimens, chemical measurements, and repeated dives. This is why the 1977 discovery still matters, not because it was the first time humans encountered darkness, or pressure, or strange animals, but because it revealed that the deep seafloor could host dense life built on chemical reactions. It gave science a new way to ask where life can be abundant, on Earth and perhaps elsewhere. The animals themselves did not become different when humans discovered them. Tube worms were already extending plumes into flow. Mussels were already housing bacteria. Shrimp were already crowding chimneys. Microbes were already converting chemicals into biomass. The discovery changed human knowledge, not the ecosystem's reality. This is worth remembering, because exploration narratives often make the world seem as if it begins when people arrive. Around underwater volcanoes, human arrival is brief and late. The vents existed in darkness before the first submersible light crossed them. They continued after the vehicle left. Science enters as observation, not ownership. The best exploration writing should keep that proportion intact. Humans are present as visitors, interpreters, and sometimes threats. The habitat remains the center. The surface of a vent mussel shell may carry its own small world. Bacteria, small grazers, sediment grains, and chemical films can gather on it. A juvenile animal may shelter between shells. A predator may search along the edge. Biogenic structure, meaning structure made by living organisms, is important in many ecosystems. Coral reefs are the obvious shallow example, but vent mussels and tube worms also create habitat by their bodies. Their presence changes flow and provides surfaces. This means the loss of large foundation species can affect smaller species that depend on the architecture they create. A tube worm colony is not only many worms. It is a thicket, a baffle, a set of crevices, a chemical interface, and a refuge. In the deep sea, where hard, complex structures can be limited, bodies become landscape. Some vent communities are dominated by animals that rely on endosymbionts, bacteria living inside tissues. Others include abundant epibionts, microbes living on outer surfaces. This distinction changes how animals feed and how they interact with the environment. A host with internal bacteria must move chemicals into its body. A host with external bacterial growth may graze, cultivate, or maintain surfaces where bacteria grow. The language of farming is sometimes used, but it should be handled cautiously because animals are not planning agriculture. They are behaving in ways that maintain access to microbial food or partners. A shrimp cleaning its appendages or scraping bacterial growth is not a farmer in the human sense, but the result can still resemble cultivation, animal behavior sustaining a microbial resource that then sustains the animal. The vent field can seem crowded with specialists, yet generalists also matter. Mobile predators and scavengers may move between vent and non-vent habitats. Some bacteria can exploit a range of chemical conditions. Some animals may use vents opportunistically rather than exclusively. Drawing the boundary between vent endemic, vent-associated, and occasional visitor can be difficult, especially when observations are limited. This difficulty is not just a naming problem. It affects how scientists understand carbon flow, conservation value, and community resilience. A species found only at vents may face different risks than a species that feeds there but reproduces elsewhere. The camera may show them in the same frame, a crab beside a worm beside a fish, but their dependence on the vent may be very different. Seeing animals together is the beginning of ecology, not the end of it. There are moments in vent footage when nothing much happens. The plume rises. A shrimp shifts. A crab pauses. Particles drift. The machine hovers. These quiet intervals are not empty. They show maintenance, the part of life that rarely becomes a highlight. Most animals spend more time feeding, holding position, sensing, and conserving energy than they do fighting or escaping. This is especially true in the deep sea, where unnecessary movement can be costly and opportunities may be patchy. A good vent script should leave room for that stillness. The alien life around underwater volcanoes is not always doing alien things. Often it is doing ordinary animal things in a place where ordinary animal things require unusual biology. It waits. It feeds. It adjusts. It survives another minute in the flow. The pressure of the water column also changes the relationship between heat and life. At the surface, water at 400 degrees Celsius would be steam under ordinary pressure. At vent depths, high pressure keeps hydrothermal fluid in a different state as it emerges, allowing extremely hot fluids to move as liquid or supercritical like mixtures depending on conditions. This lets chemical reactions occur in ways that would be impossible in a shallow open pot of water. Metals remain dissolved until mixing triggers precipitation. The plume becomes a chemical event shaped by pressure as much as temperature. The animals live outside the hottest core, but their habitat depends on these pressure-altered fluids. The deep ocean does not simply hide a surface like volcano underwater. It creates a different volcanic chemistry, one that belongs to depth. The animals around vents often have limited tolerance for ordinary surface conditions, which makes aquarium display and laboratory study difficult. Some require high pressure, low temperature, specific chemistry, or microbial partners that are hard to maintain. Researchers have developed pressure vessels and specialized systems to keep certain organisms alive long enough for experiments. But many vent animals cannot be studied like common shallow water species. This limitation shapes what is known. Behaviors easy to observe in coastal aquaria may remain poorly known for vent animals. Reproductive events may be missed. Long-term physiology may be inferred from fragments of evidence. The difficulty is not a failure of curiosity. It is a reminder that these animals belong to conditions our laboratories must struggle to imitate. Their normal world is an extreme environment for our tools, even when it is simply home for them. A hydrothermal vent also has a smell in chemical terms, even if humans do not smell it directly at depth. Hydrogen sulfide is often associated with a rotten egg odor at the surface and is toxic in sufficient concentrations. At the vent, it becomes an energy source for microbes. Methane, hydrogen, iron, and other reduced compounds can also feed microbial metabolisms depending on the system. The idea that a poison in one context becomes food in another is one of the most important shifts in understanding vent life. It is not that toxicity disappears. It is that evolution has built pathways and partnerships that use dangerous chemistry under controlled conditions. A compound has no moral character. It is harmful, useful, or both depending on concentration, organism, and context. Around underwater volcanoes, life is built from that context. When a vent field collapses after an eruption, it can look like a disaster in human terms. Dead animals, buried chimneys, bare lava, altered chemistry. But natural disturbance also creates new surfaces and new flow paths. The system is destructive and generative at the same time. This does not mean every disturbance is beneficial, and it does not excuse human damage. Natural eruptions are part of the evolutionary history of vent species. Industrial disturbance may operate differently in timing, frequency, and physical effect. Still, understanding natural disturbance helps scientists understand resilience. Which species arrive first? Which depend on mature structure? How far can larvae travel? How quickly do symbionts become available? The recovery of a vent field is not the return of a lost paradise. It is the assembly of a new community under changed conditions, using whatever living pieces can reach the new rock. Some of the smallest animals in vent communities live almost invisibly between the famous ones. Copepods, amphipods, small worms, juvenile snails, and other meiofauna occupy spaces in mats, sediment pockets, shells, and tubes. They graze, scavenge, reproduce, and become prey. These smaller organisms can be essential to understanding the whole food web, but they are easy to miss in wide camera shots. They may require suction samples, cores, careful sorting under microscopes, and genetic analysis. A spectacular ROV frame can create the impression that the community consists of a few large types. The sample tray tells another story, full of small bodies that make the ecosystem more complex. Around underwater volcanoes, alien life is not only the giant worm in the light. It is also the tiny crustacean in the shell crevice and the microbial filament on the rock. The term extremophile is often used for organisms that thrive in conditions humans consider extreme. Many vent microbes fit that term well, especially those tolerating high temperatures, acidity, pressure, or chemical stress. For animals, the term should be used with more precision. Vent animals are extremotolerant in particular ways, often through symbiosis, behavior, and physiology, but they still avoid the most extreme parts of the vent. A Pompeii worm is not the same kind of extremophile as a hyperthermophilic archaeon, living at temperatures no animal can tolerate. A tube worm depends on microbes that perform chemistry the animal cannot. The word is useful if it points towards specific adaptations. It becomes misleading if it turns every vent animal into a superhero of survival. The real story is narrower and better. Each organism has found a workable range inside a habitat full of gradients. The deeper a vent lies, the more expensive every minute of observation becomes. Ship time is limited. Weather can interrupt. Equipment can fail. A tether can snag. A manipulator can jam. Batteries and schedules impose boundaries on curiosity. Even when a dive succeeds, scientists must choose what to film, what to sample, and what to leave alone. This scarcity of access shapes the scientific record. Some vents are well studied because they are reachable and repeatedly visited. Others are known from brief surveys. Some are inferred from plumes and not yet imaged. The absence of evidence from a remote ridge may reflect lack of observation more than lack of life. The deep sea is not only biologically patchy, it is patchy in human knowledge, with bright islands of data surrounded by large areas where the map still has low resolution. The first moment of seeing a new vent field can still happen today. A camera drops toward a ridge. The sonar suggests rough terrain. The chemical sensors have found a plume. Then the lights catch a mineral wall, a shimmer, a cluster of animals. The control room quiets because everyone is trying to understand what kind of place has entered the frame. Even now, new vent fields can reveal unexpected species, unusual chemistry, or different community structure. The discovery is no longer that vents exist. The discovery is how many versions of vent life the planet has made. This keeps the subject from becoming a finished chapter. Hydrothermal vents are known enough to explain, and unknown enough to keep changing the explanation. That balance is where the best deep-sea stories live. A dead whale on the seafloor and an active vent might seem unrelated, but both show how animals respond to concentrated resources in the deep. A whale fall begins as a massive pulse of organic matter from above. Scavengers arrive, soft tissue is consumed, bones become habitat, and sulfide produced by decay can support chemosynthetic microbes and specialized animals during later stages. A vent begins with chemical energy from below, not a carcass, but it too creates dense life around a localized source. The comparison helps place vents within the hidden ocean rather than outside it. The deep sea has multiple islands of energy: carcasses, seeps, vents, seamounts, canyons, and productive slopes. Each one gathers life differently. Around underwater volcanoes, the island is built by hot water and rock chemistry, but the ecological principle of concentration remains recognizable. The intense chemical environment can also preserve and destroy evidence in different ways. Mineral deposits may entomb microbial textures or animal remains. Acidic or hot fluids may dissolve or alter materials. Shells may persist in some zones and disappear in others. Tubes can remain after the animal dies, marking former colonies. The fossil record of ancient vent communities is therefore uneven. Scientists have identified fossil hydrothermal deposits and ancient vent-related faunas, but preservation depends on mineralization, burial, and later geological history. Modern vents help interpret these ancient traces, but they are not perfect replicas of past systems. The animals have changed. The ocean has changed. The chemistry may have changed. Still, the basic link between hydrothermal circulation and chemosynthetic life gives paleontologists a way to read certain rocks as former habitats, not just mineral deposits. The vent story also belongs to the carbon cycle. Microbes fix carbon dioxide into organic molecules using chemical energy. Some of that carbon becomes animal biomass. Some is respired back to carbon dioxide. Some may enter sediments or be carried away in particles. Compared with global photosynthesis, vent carbon fixation is small, but locally it can dominate. Local dominance is what the camera sees. It does not see global budgets. It sees a chimney surrounded by animals that could not be explained by the slow rain from above alone. Scale matters again. Vents may not run the planet's carbon cycle, but they run their own neighborhoods with intensity. For the animals attached to the chimney, local chemistry is the difference between a bare rock and a crowded home. The animals around vents are not evenly distributed with depth. Depth influences pressure, temperature of background water, oxygen, larval dispersal, and regional species pools. A vent at 800 meters may have different connections to the broader ocean than one at 3,000 meters. A vent in the Western Pacific may differ from one in the Atlantic, even at similar depths, because evolutionary history and dispersal pathways differ. This is why broad statements about vent animals should be checked against location. There is no single typical vent community that represents all others. The title can be singular, but the reality is plural. Alien life around underwater volcanoes means many communities, many depths, many chemistries, many histories, and many forms of dependence on the same underlying fact: hot, altered seawater returning from the crust can feed life in darkness. A camera beam crossing a vent field may show animals arranged like a diagram without meaning to. Closest to the active orifice, surfaces may be too hot or unstable for large animals. Nearby, bacterial mats and heat-tolerant species occupy cracks. Farther out, tube worms or mussels cluster in diffuse flow. Beyond them, mobile predators and scavengers move through the edges. Then, the density fades into ordinary deep seafloor. The exact pattern varies, but zonation is common because chemical and thermal gradients organize life. The animals become a living contour map. They trace invisible lines in the water. Where one species stops and another begins, the chemistry has changed. The ROV sees biology. The biology reveals physics and chemistry. This is why vent footage can be read, not just watched. Sometimes the ROV returns to a marked site and finds that the scene has changed enough to feel personal, even though the changes are ecological. A chimney that stood upright has broken. A colony has expanded. A patch of mussels has thinned. A temperature probe is coated with growth. A marker placed in bare rock now sits beside new tubes. Long-term observation is essential because vents are not static displays. Time series studies can show growth rates, colonization, disturbance, and shifts in flow. The first dive gives a portrait. The return dives give a life history of the place. Around underwater volcanoes, the habitat itself has behavior in a loose sense. It pulses, redirects, builds, collapses, and changes the opportunities available to animals. Watching once is seeing a face. Watching repeatedly is beginning to understand expression. The animal's apparent abundance can also hide the difficulty of recruitment. A mature tubeworm cluster may contain many adults, but if larvae fail to settle for several years, the colony's future may be fragile. A mussel bed may look stable while the flow beneath it shifts. A shrimp swarm may remain dense until a sudden change forces relocation. Population health is more than the number visible in one frame. It includes age structure, reproduction, genetic diversity, connectivity, and habitat stability. This is why environmental assessments at vents require more than spectacular video. They require repeated surveys, species identification, genetic work, chemistry, and an understanding of regional networks. The deep sea can make abundance look timeless. Science has to ask whether the abundance is renewing itself or simply remaining from a period of better conditions. The mineral particles in a black smoker can eventually settle and contribute to deposits on the seafloor. Over time, hydrothermal processes can form seafloor massive sulfides, accumulations rich in metal sulfide minerals. These deposits are part of why vents interest economic geology. They also create complex hard bottom habitats. But an active sulfide mound is not just ore in waiting. It is a living, reactive structure. Hot water passes through it. Microbes colonize it. Animals attach to it. Chemistry changes across it. The same rock can be read as resource, habitat, archive, and process depending on who is looking. A responsible account has to keep all of those readings in view because reducing the vent to only one of them misses the reality of the place. The most alien thing about vents may be the way they make the interior of Earth biological. Not in a vague spiritual sense, but materially. Heat from below drives water through rock. The rock changes the water. The water feeds microbes. Microbes feed animals. Animals create habitat for other animals. Predators and scavengers move through the resulting structure. A chain begins with geology and becomes ecology. This happens without roots, leaves, daylight, or soil. It happens in pressure and darkness, on mineral towers built by fluids hotter than any animal can directly endure. The route is unfamiliar, but the outcome is recognizable: a community. Bodies crowd where food exists. Offspring search for new habitat. Species compete, cooperate, and die. The volcano does not create life by magic. It creates conditions under which life can do what life does. Imagine the camera resting just outside a shrimp swarm. The vehicle holds position, making tiny corrections with its thrusters. The chimney exhales, white bodies shift and settle. A fish appears at the edge of the beam and waits. In the background, a tube-worm plume withdraws and reopens. Nothing in this frame requires exaggeration. The temperature gradient is real. The sulfide is real. The bacteria are real. The animals are real. The uncertainty is real too, because the camera cannot show every chemical exchange, every larval journey, every microbial pathway, or every hidden cavity below the rock. That is enough. The best deep-sea images do not need to pretend the ocean is unknowable. They need only show how much there is to know and how carefully it has to be approached. Hydrothermal vents also force a distinction between survival and flourishing. An organism may tolerate a condition but not grow well in it. It may survive a temperature spike but fail to reproduce. It may persist at the edge of a vent but only flourish where flow is steady and chemical supply is balanced. This matters when interpreting extreme tolerance. The maximum condition an organism can survive is not necessarily its preferred habitat. A headline number can hide the more important range where feeding, growth, and reproduction actually happen. Around underwater volcanoes, the animals are often described by their extremes, but their real lives happen in workable averages, brief fluctuations, and behavioral adjustments. The question is not simply what they can withstand, it is where they can complete a life. The surrounding deep sea sends its own animals to the vent edges. Eelpouts and other deep-sea fishes may be associated with vent fields in some regions. Crabs and other mobile predators can link habitats. Scavengers can take advantage of dense biomass. The boundary between vent endemic and broader deep-sea fauna can be porous, especially for mobile animals. This creates a layered community. At the center are organisms tightly dependent on hydrothermal chemistry. Around them are animals that exploit the concentration of prey. Around those are species from the wider deep sea that may only occasionally enter. The vent is therefore not an isolated theater with a fixed cast. It is a feeding center, a nursery for some, a hunting ground for others, a settlement site for larvae, and a chemical source that changes the behavior of animals beyond the most obvious zone. The animals can be harmed not only by removing them, but by changing flow. A chimney's structure directs hot fluid. A crack supplies diffuse warmth. A mussel bed may depend on seepage through the substrate. If rock is broken, covered, or compacted, the chemistry reaching the animals can change even if some animals remain in place. This is one reason physical disturbance at vents can have biological effects that are not visible immediately. The habitat is a system of flow paths, not only surfaces. A machine might leave behind what looks like hard seafloor, but if the plumbing has been altered, the living conditions may be gone. In vent ecosystems, habitat includes the movement of water through rock. Protecting the visible animals without protecting the invisible flow would miss the mechanism that keeps them alive. The comparison to Europa or Enceladus should remain restrained, but it is still valuable. These icy moons may contain subsurface oceans, and tidal heating or water-rock reactions could create chemical gradients. On Earth, hydrothermal systems show that chemical gradients can support microbial life and, through microbes, larger communities. The analogy does not predict animals elsewhere. Complex animals require long evolutionary histories, ecological stability of certain kinds, and many conditions not guaranteed beyond Earth. But microbial life, if it exists elsewhere, might use energy sources that do not involve sunlight. Vents make that idea concrete. They show dark water, rock chemistry, and microbial metabolism supporting visible life. They do not answer the question of alien oceans, but they make the question less fanciful and more scientific. The animals around vents are not ancient survivors unchanged from the beginning of life. They are modern species in modern lineages, adapted to habitats that have existed in some form through deep time. This distinction prevents a common mistake. A tube worm is not primitive because it lacks a mouth as an adult. That condition is specialized. A vent shrimp is not a relic from another age because it has reduced eyes. It is adapted to its own sensory environment. Evolution does not always move toward complexity in the way people imagine. It moves toward workable forms under current pressures. Losing a structure can be as evolutionary as gaining one. Around underwater volcanoes, some of the most striking features are reductions, partnerships, and dependencies. They are not signs of being unfinished. They are signs of being precisely shaped by a habitat, unlike the one humans know. A scientist watching a vent monitor may recognize a species by posture before details are clear. The way a shrimp clusters, the shape of a tube, the outline of a mussel bed, the movement of a crab. Then, the camera comes closer, and the identification may change. What seemed like one species becomes another. What seemed like mineral coating becomes bacterial mat. What seemed empty contains juveniles. Deep sea observation often begins with correction. The first impression is useful, but not final. Taxonomy at vents can require specimens, microscopy, DNA, and comparison with previous records. Many animals cannot be confidently named from video alone. This uncertainty is not a weakness in the scene. It is part of its honesty. The deep sea is not there to be recognized instantly. It has to be approached, sampled, checked, and sometimes revised. The water flowing from a vent has traveled through a hidden path. It began as seawater, entered the crust, warmed, reacted, lost magnesium, gained metals and reduced compounds, then rose back through fractures. The journey may take years or longer depending on the system. By the time the fluid exits the chimney, it is no longer ordinary seawater. It carries a history of contact with rock, heat, and pressure. That history becomes visible as black particles, chimney growth, microbial metabolism, and animal bodies. Every plume is therefore a return. Water that entered the crust comes back altered enough to feed a world. The animals do not see the journey, but their lives depend on it. A worm plume reaching into diffuse flow is touching the end of a path that began below the seafloor, in darkness deeper than the dark water itself. The ship above a vent field may drift over kilometers of water without any visible sign of what is below. There is no steam on the surface, no discoloration, no smell, no sound that tells the crew a black smoker is active beneath them. Only instruments and coordinates connect the deck to the chimney. This invisibility is one reason the deep sea was able to keep vents outside human knowledge for so long. The ocean's surface hides not only animals, but processes, eruptions, circulation, chemical exchange, mineral formation, and dense communities. A calm horizon can sit above a volcanic landscape. The surface does not lie, but it does not tell the whole truth. It is one layer of a planet that continues downward through water, darkness, rock, heat, and life. Vent fluids can be acidic compared with seawater, and mixing with cold seawater drives rapid mineral precipitation. The chemistry can alter not only the rocks, but also the local environment animals experience. pH, sulfide, oxygen, metals, and temperature can all vary sharply. This is why vent animals cannot be understood by depth alone. Two animals at the same depth may live in completely different chemical worlds depending on how close they are to flow. Even within one colony, individuals may experience different conditions based on position. A tube worm at the edge of a cluster may not receive the same sulfide supply as one near the center. A shrimp on one side of a chimney may experience different flow than a shrimp only a few centimeters away. At vents, small distances matter because chemistry is written in fine scale. The death of a vent community can be quiet enough that no one records it. If flow stops between expeditions, the next camera may arrive to find empty tubes and dead shells. The event itself may have happened in darkness without witnesses. This is common in deep-sea science. The record is often a before and after, with the crucial moment missing. Researchers infer from remains, chemistry, and new flow patterns. Long-term observatories help reduce these gaps by leaving instruments in place, measuring temperature, chemistry, seismicity, and sometimes imagery over time, but they are expensive and limited. Most of the deep sea still changes without an audience. Around underwater volcanoes, life can bloom, fail, and reorganize in the intervals between human visits, leaving clues for the next descent to interpret. A small juvenile tubeworm on fresh rock may be more important than a large adult in understanding the future of a field. The adult proves that suitable conditions existed. The juvenile proves that settlement is happening now, or happened recently. Juvenile stages help scientists understand recruitment, growth, and recovery. They can reveal whether a disturbed site is being recolonized or whether an aging community is failing to replace itself. Identifying juveniles can be difficult because young animals may look different from adults and may require close examination. The deep sea often hides its future in small forms. A tiny tube on basalt, barely noticeable beside a black smoker, may be the beginning of a colony that will later dominate the frame. Or it may be one of many attempts that fail when flow shifts. Vent organisms can have remarkable growth rates compared with many deep-sea animals, but those rates depend on energy supply and species. Giant tubeworms, for example, can grow quickly when conditions are favorable because their symbionts provide abundant nutrition. Other vent animals may grow more slowly. Some deep-sea corals on volcanic substrates can live for long periods and grow slowly without relying on vent chemistry. It is important not to generalize one life history across all animals around underwater volcanoes. The habitat includes fast-growing opportunists, long-lived suspension feeders, mobile predators, microbial mats, and tiny animals with poorly known timelines. The deep ocean's clocks overlap here again. Fast chemical reactions build chimneys. Rapid colonizers exploit new flow. Slow animals occupy old, hard surfaces. The volcano brings them into proximity without making their lives run at the same speed. If the ROV settles on the seafloor away from the vent and turns toward the plume from a distance, the scene can look almost peaceful. The black smoker rises against darkness. Animals cluster at its base. The plume bends slightly with current. There is no explosion, no glowing lava, no visible violence. The danger is in chemistry and change rather than spectacle. The water is hot. The sulfide is toxic. The pressure is immense. The habitat can fail. But the animals are not living in panic. They are living in routine. This calm is important. It prevents the vent from becoming only a stage for danger. The real scene is quieter. A dangerous place made usable by adaptation. A crowded community built around a flow that has become ordinary to its residents. The study of vents also depends on naming and comparing regions. The East Pacific Rise is fast spreading, with vent fields that can be disturbed by eruptions and recolonized. The Mid-Atlantic Ridge spreads more slowly and hosts different communities, often with shrimp-dominated systems. Back arc basins and volcanic arcs in the Western Pacific can have their own chemistries and species. Indian Ocean vents have revealed distinctive animals such as the scaly foot gastropod. These regional differences are not side details. They show how geology and biogeography combine. If the same basic energy source produces different communities in different oceans, then the vent is both a universal phenomenon and a local experiment. Earth keeps asking the same question in different places. What can life do with rock-powered chemistry here? The animal's symbiotic bacteria may also vary by region and host. Even when two animals appear similar, their microbial partners can differ in identity, function, or acquisition. Some hosts may contain multiple symbiont types that use different chemical pathways. Others may rely on a dominant partner. Symbiont communities can shift with environmental conditions or developmental stage. This adds another layer to biodiversity. A vent animal is not only one species, it is a host plus a microbial community, and the partnership may be essential to its ecological role. Conservation and biology therefore have to consider relationships, not just bodies. Protecting a tube worm without the conditions that maintain its symbionts would be meaningless. Understanding a shrimp without its microbial associates would be incomplete. Around vents, the individual animal is often a small ecosystem moving through a larger one. The footage can also mislead by compressing scale. A close-up of a shrimp may fill the screen and seem large. A wide shot of a chimney may shrink animals into texture. Without a reference, the viewer may lose sense of size. ROV lasers, often set a known distance apart, help scientists measure objects in the frame. A pair of green dots on a chimney or animal can turn a beautiful image into usable data. The lasers look artificial because they are, but they make the scene more honest. They reveal whether a tube is a few centimeters or much larger, whether a fish is juvenile or adult, whether a chimney wall has grown between visits. In the deep sea, scale is not obvious. It has to be brought down with the lights, measured carefully, and attached to the image before the image can become evidence. The heat at vents comes from a planet that is cooling slowly over billions of years. But the local expression of that heat can be sudden. A crack opens. A flow strengthens. A chimney begins. Animals settle. A community forms. Then the flow shifts again. Plate tectonics gives the long-term setting, but local hydrothermal plumbing controls the immediate fate of the animals. A field may sit on a ridge segment shaped by millions of years of spreading, while a tube worm's survival depends on whether warm diffuse flow continues this month. This contrast between geological duration and biological immediacy is one of the vent's quiet tensions. The forces are vast. The living margins are small. A worm does not need the ridge to last forever. It needs its crack to stay alive long enough for feeding, growth, and reproduction. When a vent field is mapped, scientists may give names to chimneys, mounds, or zones. These names help organize dives and data, but the animals do not live in named places. They live in gradients and structures. A named chimney may have several microhabitats, each with different flow. A named field may contain active and inactive areas, dense colonies and bare rock, surface life and hidden cavities. Naming is useful because it lets humans return, compare, and communicate. It should not make the habitat seem simpler than it is. The deep sea resists neat labels. A single coordinate can contain heat, cold, toxic fluid, oxygenated water, minerals, bacteria, worms, shrimp, predators, empty tubes, and the possibility of tomorrow's shift in flow. The name is a handle. The place is more complicated. Vent communities can also influence mineral deposition. Biological surfaces can affect local flow and precipitation. Microbial activity can alter chemical microenvironments. Tubeworm tubes and mussel shells can trap particles and create surfaces for further colonization. The animals are not the main architects of sulfide chimneys in the way corals build reefs from calcium carbonate skeletons, but they do interact with the mineral environment once it exists. This interaction makes the habitat partly reciprocal. Geology creates conditions for life, and life modifies the small-scale structure of the geological surface. A chimney covered in animals is not the same physical object, in ecological terms, as a bare chimney. It has become textured by bodies, and those bodies change how other organisms experience the flow. The vent can appear timeless in a single shot because the camera freezes it into composition: chimney, plume, animals, darkness. But the real system is moving at multiple speeds. Fluid exits rapidly. Particles precipitate in seconds. Microbes divide. Animals feed and shift. Larvae drift for days or weeks. Colonies develop over years. Ridges spread over millions of years. To understand the scene, all of these timescales have to be held together. A single frame contains processes that began below the seafloor long before the vehicle arrived, and processes that will end moments after the water mixes. The animal visible in the light is the meeting point of fast chemistry and slow evolution. This is why vent footage rewards patience. The longer one looks, the more movement appears inside what first seemed like a fixed landscape. Human language often reaches for metaphor at vents because the direct facts are unfamiliar. Chimneys smoke. Worms form forests. Shrimp swarm like snow. White towers look like castles. But each metaphor has to be set down before it takes over. The smoke is mineral particles. The forest is animal tubes. The snow is moving bodies or marine particles. The castle is carbonate or sulfide built by water-rock reactions. The real descriptions are stronger because they keep the habitat attached to evidence. A metaphor can open a door, but science has to walk through it. Around underwater volcanoes, the world already looks like imagination. The responsibility of narration is to bring it back to the physical: temperature, pressure, sulfide, oxygen, basalt, bacteria, shell, tube, claw, plume. A claw mark on a tube, a broken shell, or a cleared patch in a microbial mat can tell a small story without showing the event. Predation may be inferred from damage. Grazing from scraped surfaces. Collapse from broken mineral fragments. Settlement from tiny juveniles. Much of ecology is reading traces, especially in habitats where direct observation is brief. The deep sea does not always show the moment, but it often leaves consequences. Those consequences must be interpreted cautiously. A broken shell may have been crushed by a predator, by falling rock, by sampling equipment, or by natural decay. Evidence needs context. The best vent science combines the patience of watching with the skepticism of checking. It notices the trace, asks what could have made it, and resists turning the first possible explanation into certainty. Hydrothermal vents can affect surrounding seawater chemistry beyond the immediate habitat, contributing metals and other substances to the deep ocean. The global significance of these fluxes depends on the element, the rate of venting, and how particles settle or remain suspended. For biology, the local effects are often most visible, but geochemists study vents as part of ocean-wide cycles. Iron from hydrothermal sources, for example, can be transported in the deep ocean under certain conditions and may influence microbial processes far from the vent. This widens the story without turning it into exaggeration. The animals live close to the chimney, but the plume may enter larger circulation. The vent is a small place with chemical connections extending outward. A black cloud in the camera beam is also part of the ocean's hidden chemistry. If a vent field is mined or physically removed, the immediate loss would be visible. Chimneys broken, animals crushed or displaced, plumes of sediment or debris, hard substrate altered. The less visible losses could be just as important: disrupted flow paths, lost microbial habitats, broken population connections, and removal of structures that took years or longer to form. Predicting recovery is difficult because vent systems vary, and because long-term industrial disturbance has not occurred at the scale proposed in some discussions. Natural analogs help, but do not answer every question. The uncertainty is not a reason to assume safety. In fragile or poorly understood systems, uncertainty can be a reason for restraint. Around underwater volcanoes, where life is tied to the exact structure and chemistry of the seafloor, damage cannot be evaluated only by how much rock remains. There is also a human emotional response to vents that should not be dismissed, but it should be kept proportionate. Seeing dense life in darkness can feel like finding a hidden room in the planet. The scene is beautiful, strange, and humbling. But the animals are not there to make humans feel awe. They are feeding, growing, competing, and reproducing. Their lives have their own terms. The best use of awe is to slow attention, not to inflate the subject. It should make the viewer more careful with facts, not less. A tube worm does not become more remarkable when called impossible. It becomes more remarkable when its possibility is explained. No mouth, internal bacteria, sulfide and oxygen carried by hemoglobin, carbon fixed in darkness, a body built at the edge of volcanic flow. The seafloor sometimes reveals inactive chimneys before active ones, like ruins leading toward a living settlement. The ROV crosses mineral mounds with few animals, then dead tubes, then faint shimmering water, then stronger flow and dense life. This approach can show the spatial history of hydrothermal activity. Fluids moved here once. They moved there later. They may be strongest now at the chimney ahead. Mapping these transitions helps researchers understand how fields evolve and how animals track changing flow. The vent is not a single dot of activity. It is a landscape with memory. Some of that memory is geological, written in mineral deposits. Some is biological, written in shells, tubes, and the distribution of living animals. The ROV reads both as it moves. Vent animals must also manage waste products from their own metabolism and from their symbionts. A partnership that brings food also creates chemical byproducts that must be transported, transformed, or excreted. Blood chemistry, tissue structure, and microbial balance all matter. These internal processes are less visible than plumes and tubes, but they are essential to the animal's success. The host is not simply a container filled with helpful bacteria. It is an active physiological environment. Symbiosis works because the host and microbes regulate exchange across boundaries inside the body, just as the community regulates life across boundaries in the water. The vent repeats the same theme at different scales, mixing without losing control. Oxygen and sulfide in the water. Host and microbe in tissue. Heat and cold at the chimney. Life depends on contact, but also on separation. The chemistry of a vent can shift as seawater-rock reactions proceed and as pathways through the crust open or seal. A fluid sample taken 1 year may differ from a sample taken later at the same site. Temperatures can rise or fall. Metal concentrations can change. The relative importance of sulfide, hydrogen, methane, and other compounds can vary. Animals respond to these shifts through distribution, growth, and survival, but the response may lag behind the chemistry. A colony can remain where conditions have begun to decline, especially if the animals are attached. This makes vents difficult to interpret from a single visit. A thriving community may reflect current conditions or the recent past. A sparse area may be newly active or unsuitable. Time is needed to know which. The most extreme parts of the vent are often lifeless to animals. This is important because it corrects the fantasy that life simply becomes more abundant as conditions become harsher. The central jet of hot fluid may be dominated by chemistry and microbes adapted to high temperatures, while animal life gathers where the fluid is diluted. Life has limits, and those limits differ between microbes and animals. A hyperthermophilic microbe may survive temperatures that would destroy animal proteins. An animal may survive near those conditions only by remaining in cooler mixtures. The vent field is therefore a layered display of biological possibility. Microbes approach the extreme more closely. Animals build around the moderated edge. The visible community depends on the invisible organisms that can work nearer the chemical source. Hydrothermal vents are sometimes separated into high-temperature focused flow and lower-temperature diffuse flow, but the distinction is part of a continuum. Focused flow exits through chimneys or orifices. Diffuse flow seeps through cracks, porous rock, or biological structures after mixing below the surface. Diffuse flow can be especially important for animals because it provides chemicals at temperatures they can tolerate. A field of tube worms or mussels may depend less on the dramatic black plume than on warmer, gentler seepage around it. The camera loves the smoker because it moves like a signal. The animals may be living from the quieter leaks. Understanding vents means looking not only at the tallest chimney, but at the subtle shimmer between tubes, the pale matte on rock, the warmth rising where no plume dominates the frame. When the word volcano appears in the title, it can imply lava. But hydrothermal vent life does not require animals to live on erupting lava. It requires heat, fluids, and chemical gradients, often associated with volcanic or tectonic activity. Lava may create the fresh substrate and fractures. Magma or hot rock may provide heat. But the animals live after water has circulated and returned, not in direct contact with molten rock. This distinction keeps the scene scientifically grounded. The animals are not surviving lava, they are surviving hydrothermal conditions created by volcanic systems. The underwater volcano is important because it supplies heat and structure. The living habitat is the place where that heat has been translated through water and rock into chemistry that biology can use. A vent field may sit in complete darkness, but it is not quiet chemically. Electrons move through microbial metabolisms. Carbon dioxide becomes organic carbon. Sulfide becomes sulfate. Hydrogen is oxidized. Methane may be transformed. Minerals precipitate. Oxygen is consumed. These processes are not visible as actions, yet they build every visible animal in the frame. A crab's muscle, a shrimp's shell, a worm's tube, a muscle's tissue, all are connected to chemical transformations happening at microbial scales. The scene teaches a reversal of attention. Instead of looking at the largest animal and asking what it eats, we look at the smallest organisms and ask how their reactions make large animals possible. Around vents, biology grows upward from chemistry so directly that the usual surface categories feel incomplete. At the end of a long dive, the cameras may pass one last cluster of animals almost accidentally. A few white tubes on the edge of a rock, a crab half hidden in shadow, a small patch of bacterial mat glowing pale under the lamps. These minor scenes matter because they show that vent life is not only the central chimney, it spreads into edges, cracks, and transitional zones. It fades gradually into surrounding deep sea. It leaves traces where flow used to be, and small beginnings where flow has just appeared. The alien life around underwater volcanoes is therefore not one spectacle. It is a habitat with margins, histories, failures, and quiet places. It contains the dramatic plume, but it also contains the small animal under the shell and the thin microbial film on the cooling rock. If the ROV carries a suction sampler, it can collect small animals from mats and crevices that the camera cannot resolve. The sample may look unimpressive on deck, a container with sediment, fragments, and tiny moving or preserved bodies. Under a microscope, it becomes a community. Meiofauna and small macrofauna help reveal diversity, trophic links, and reproductive stages. They may include juveniles of larger animals or species that never become large. Without these samples, the vent ecosystem would be biased toward what the lens can easily see. This is a recurring issue in deep-sea exploration. The camera gives atmosphere and context. The sample gives detail. Neither replaces the other. The living reality is larger than both, but together, they let science move from astonishment toward understanding. The animals around vents are shaped not only by what they need, but by what they must avoid. Too much heat, too much sulfide, too little oxygen, unstable surfaces, predators, competition, loss of flow, burial by lava or sediment. Evolution works with both opportunity and constraint. The opportunity is chemical energy in a food-poor deep sea. The constraints are the narrow conditions under which that energy can be used safely by animals. This is why vent adaptations often seem so precise. A plume that can withdraw. A body that houses bacteria. A behavior that maintains position near flow. A larval strategy that searches for rare habitat. Each adaptation answers a problem created by the same system that provides food. The volcano gives, but it does not give gently. The discovery of vents also altered how scientists think about the limits of the biosphere within Earth's crust. Microbial life has been found in many subsurface environments, and hydrothermal circulation provides pathways for energy and materials below the seafloor. Vent systems are part of this larger deep biosphere discussion. Animal life beneath vent crust, where observed, is a much more specific finding than microbial life in the subsurface. It requires space, suitable temperature, chemistry, and connection to the seafloor or fluids. Still, the presence of animals under lava plates near vents expands the habitat vertically in a way that would have seemed unlikely not long ago. The rock is not only under the animals. In some places, it is around them, above them, and part of the route by which their young may move. The ocean above vents can carry hydrothermal signals far from their source. Scientists may detect elevated methane, manganese, iron, temperature anomalies, or particle loads in plume surveys. These traces can point toward undiscovered vents, especially when combined with seafloor maps and current models. Searching for vents can feel like following smoke without seeing fire, except the smoke is dissolved chemistry and mineral particles in black water. The ship crosses lines above the seafloor. Instruments rise and fall. Data appears as numbers and plots before it becomes an image. The eventual camera view of a chimney is dramatic because it gives the signal a source. The plume stops being an anomaly and becomes a place, a crack, a tower, a community, and animals using chemistry that had first appeared only as a change in a sensor reading. The ship's deck after a dive can smell of seawater, oil, metal, and samples, but not truly of the vent as it exists below. Fluids have cooled, gases have escaped or oxidized, animals have been removed from pressure. Mineral fragments sit in trays under lights that are too bright and air that is wrong for them. Yet this transition from seafloor to deck is where much scientific work begins. Samples are labeled, photographed, preserved, frozen, or transferred to experiments. Metadata is checked. Time, depth, location, temperature, and visual context are matched to each object. The care in these ordinary actions matters because a sample without context loses much of its meaning. A worm from a vent is not just a worm. It is a worm from a particular flow, a particular depth, a particular colony, at a particular moment in the life of a changing field. Some words used for vents can carry hidden assumptions. Primitive, bizarre, monster, living fossil. Impossible, these words may attract attention, but they often make the biology less accurate. Vent animals are not primitive simply because their habitat is extreme. They are highly specialized modern organisms. They are not monsters because they look unfamiliar. They are not impossible because they have been observed, sampled, and explained. The better language is more specific, symbiotic, chemosynthetic, endemic, pressure-adapted, sulfide-tolerant, thermally constrained, microbe-dependent. Specific words may seem less dramatic, but they create a clearer kind of wonder. They tell us what is actually strange. Around underwater volcanoes, the truth does not need to be enlarged. It needs to be focused. A vent community may contain both cooperation and conflict in the same square meter. Tube worms and bacteria depend on each other. Mussels provide habitat for smaller animals while competing for space. Grazers consume microbial films that also support other organisms. Predators feed on animals that depend on symbionts. Ecology is rarely one mood. The same structure can shelter one species and expose another. The same flow can feed one organism and poison another. The same eruption can destroy an old community and create fresh habitat for a new one. This is why vents should not be described only as harsh or only as generous. They are both, depending on the organism, the moment, and the distance from flow. The animals live in that mixture, rather than in a simple story of survival against darkness. The dark water around the vent also protects it from ordinary human intuition. People are used to ecosystems with visible foundations: grass, leaves, algae, coral, trees. At vents, the foundation is mostly invisible chemistry and microbes. That makes the habitat easy to underestimate if the large animals are absent or if the camera misses the active flow. A bare-looking rock with diffuse seepage may be biologically important. A pale matte may be active primary production. A small crack may supply a colony hidden behind the chimney. The deep sea asks observers to look for evidence that does not always resemble abundance. Life around underwater volcanoes can be spectacular, but it can also be subtle, and both forms deserve attention. Hydrothermal systems can also vary in longevity. Some venting may persist for decades or longer in a given area, while individual chimneys can change much more quickly. The lifespan depends on geological setting, heat supply, permeability, mineral clogging, and tectonic activity. For animals, the relevant lifespan is the duration of suitable habitat, not the duration of the broader ridge system. A field that seems long-lived to a larva may be brief to a geologist. A chimney that persists for years may be enough for rapid-growing species, but not for slow colonizers. Time at vents is therefore relational. Long and short depend on whose life is being measured. The volcano does not keep time for the worm, but the worm's life is still written in the volcano's changes. There is a moment after the ROV leaves the bottom when the plume is gone but the particles remain. They drift in the lights as the vehicle rises, and for a while it is impossible to know whether they came from the vent, the seafloor, the water column, or the vehicle's own disturbance. This ambiguity is typical of deep-sea work. Evidence moves. Context matters. A particle is not just a particle until it is measured, located, and interpreted. The ocean does not label its clues. Scientists must build meaning from position, chemistry, images, and repeated patterns. Around underwater volcanoes, even the haze in the water can become part of the evidence, but only after it is treated with patience rather than assumption. The fact that animals can grow densely in darkness without photosynthetic plants does not make plants unimportant. It reveals another route. Surface ecosystems taught biology one major pattern: sunlight to photosynthesis to food webs. Vents added another: geological chemistry to microbial metabolism to food webs. Both are natural, both obey energy limits, both depend on organisms that fix carbon. Both can support animals that then compete, prey, reproduce, and die. The difference is not life versus non-life, or normal versus alien. The difference is the source of usable energy. Once that energy is captured, ecology begins to look familiar again, even when the animals do not. A crab still eats. A worm still grows. A larva still searches. The pathway into the food web is unusual. The pressures inside the food web are recognizable. It is also possible to overstate how separate vent communities are from one another. Some species disperse widely, and some ridges provide stepping stones of habitat. Other species show strong regional separation. Both patterns exist. Connectivity is a central question because it affects evolution, recovery, and conservation. If populations are well connected, a disturbed site may receive larvae from elsewhere. If they are isolated, loss may be much harder to reverse. Genetics can reveal relationships that geography alone cannot. Two fields far apart may be connected by currents, while two closer fields may be separated by barriers in flow or larval survival. In the deep ocean, distance is not just kilometers, it is currents, time, larval biology, chemistry, and chance. The organisms at vents are part of evolutionary lineages that adapted repeatedly to chemosynthetic habitats. Some groups also occur at cold seeps, whale falls, or other reducing environments, suggesting that transitions between these habitats may have occurred over evolutionary time. Whale falls, for example, may provide sulfide-rich conditions during bone degradation that resemble some aspects of seep or vent habitats for certain organisms. This does not mean whale falls are vents, or that every vent animal uses whale falls. It means the deep sea contains multiple reducing habitats that may act as evolutionary stepping stones for some lineages. The hidden ocean is connected by chemistry as well as currents. A worm lineage may find opportunity wherever reduced compounds, microbes, and suitable surfaces come together, even if the physical setting differs. Around a vent, a dead animal is not wasted. A fallen shrimp can be eaten by another shrimp, a crab, a fish, or small scavengers. Microbes break down what remains. Shells and tubes persist as structure. Nutrients return to the system. This recycling is ordinary ecology, but the speed and density can feel different because so many animals are concentrated in a small zone. Death is also part of how scientists see stress. Empty shells, decaying tissues, and abandoned tubes can indicate changing conditions. A vent field is not only a place where life appears in impossible abundance, it is a place where bodies enter cycles quickly, where the energy that came from chemistry passes through tissue and returns to chemistry again. The underside of a tubeworm colony can be more complex than the top. Tubes overlap, twist, and create spaces where small animals shelter. Warm water may move through the cluster differently than it moves across bare rock. The colony changes the flow that feeds it. Engineers might call this feedback. Ecologists might call it habitat modification or facilitation. The language matters less than the process. Once animals establish, they alter the environment for others and sometimes for themselves. This means the vent community is not only a response to geology, it becomes a biological structure with its own internal conditions. The volcano opens the possibility, but the animals build texture inside that possibility, turning a crack in rock into a thicket of living surfaces. The fragility of vent communities is not always visual. A tubeworm tube can look sturdy, a mussel shell can look armored, a chimney can look like rock that could withstand anything. But the living system depends on flow that may be redirected by small physical changes. Breaking a chimney, covering a crack, or altering circulation can remove the chemical conditions without immediately removing every visible structure. This is why habitat protection at vents requires understanding function, not only appearance. A protected area drawn around visible animals may miss source flows or subsurface pathways. A survey that records only large organisms may miss microbial foundations. Around underwater volcanoes, what is invisible can be as important as what is filmed. Heat, chemistry, microbes, and flow are the habitat. As the camera passes along a chimney wall, it may find a seam where black mineral gives way to white bacterial growth, then to red plumes, then to bare rock. The scene changes over less than a meter. This fine-scale patchiness is one reason vents are such valuable natural laboratories. They allow researchers to study how organisms respond to steep gradients over distances small enough to measure directly. An animal at one spot may experience a different world from an animal an arm's length away. In most human landscapes, a meter is a small distance. At a vent, it can cross from too hot to habitable, from chemically rich to poor, from crowded life to bare mineral. The volcano writes ecology in close handwriting. The discovery of chemosynthetic ecosystems did not replace the importance of photosynthesis in the ocean. Most ocean life still depends directly or indirectly on sunlight-driven primary production. Vents are exceptional in their source of local energy, not in being globally dominant. Keeping that proportion is important. The value of vents does not depend on overstating their size or global productivity. Their significance lies in showing that a different kind of energy system can support complex communities, in revealing unusual adaptations, and in connecting geology to biology. A small place can change a large idea. The first vent communities occupied limited patches of seafloor, but they changed the conceptual map of life. They showed that the deep ocean was not only a place where surface life falls downward, but also a place where Earth feeds life upward. If a vent field is compared with a city, then microbes are not simply the workers at the bottom. They are also the power system, the farms, the chemical factories, and in some cases the organs inside the citizens. The comparison quickly breaks, but it points toward how deeply microbial life is woven into everything visible. Animals at vents are often holobionts, host organisms plus microbial partners, functioning together in ways that shape survival. This does not erase the animal. A tube worm is not merely a container for bacteria. It has anatomy, behavior, development, and evolution of its own. But it cannot be understood without its symbionts. Around underwater volcanoes, individuality becomes layered. A single body may contain an entire chemical partnership, and a single colony may contain thousands of such partnerships standing in the flow. The surface world often treats darkness as absence. At vents, darkness is just the normal condition in which other senses and processes matter. The animals do not wait for light to begin their lives. Microbes do not require a dawn. Fluids do not stop moving because no eye is watching. This makes the ROV's artificial light both helpful and intrusive. It lets humans see the scene, but it does not define the scene. The real vent is a chemical and physical habitat before it is a visual one. To understand it, the viewer has to imagine the lights turned off and still know that the community remains: plumes extended into water, shrimp clustered by flow, bacteria fixing carbon, crabs moving by touch and chemistry, the chimney building itself in the dark. A vent field can also be a place of sharp mortality for larvae. The same chemical cues that indicate habitat might lead to a site already overcrowded, unstable, or declining. A larva may settle where flow later stops. It may fail to acquire the right symbionts. It may be eaten before maturity. Recruitment is therefore both opportunity and risk. Scientists rarely see these failures directly because larvae are small and the deep sea is vast. But the structure of adult populations, juvenile abundance, and genetic patterns carry hints of success and failure. Every dense adult colony represents a filtered outcome. Many beginnings did not survive long enough to appear in the camera beam. Some vent fields have been incorporated into marine protected areas or management discussions because of their ecological uniqueness and vulnerability. The details differ by region and jurisdiction, and governance of areas beyond national jurisdiction remains complex. The difficulty of managing deep sea vents comes partly from their remoteness and partly from their connectivity. A field may lie far from shore, but still be part of a ridge network. Species may disperse beyond political boundaries. Mineral interests may overlap with scientific and conservation values. Effective protection requires maps, baseline data, monitoring, and international cooperation. The animals around underwater volcanoes cannot advocate for the conditions that keep them alive. Their protection depends on whether humans can translate deep sea knowledge into restraint before extraction becomes routine. The final image of a vent does not have to be the largest chimney. It might be a small leak in the rock, a shimmer that barely distorts the water, with a few animals gathered around it. That small leak contains the whole principle. Seawater has entered the crust, reacted with rock, warmed, returned, and made chemistry available to microbes. Microbes have turned that chemistry into food. Animals have found the microbes, partnered with them, grazed them, eaten the grazers, or built homes beside them. The scale is modest, but the idea is immense. A crack in volcanic rock can become a living place in a part of the ocean where food is usually scarce. That is the story the camera keeps returning to, whether the frame contains a towering smoker or only a pale film moving in the dark. The camera pulls back one last time. The chimney is still there, exhaling into black water, but the animals have become small enough to look almost like texture on the rock. A crab crosses a white tube. Shrimp hold themselves near the flow. Mineral particles rise and disappear above the lights. Nothing here is outside nature. The volcano leaks heat through cracks, the water carries chemistry out of stone, the microbes make food from reactions, and the animals gather where that work can sustain them. Then, the ROV turns, and the beam leaves the chimney behind. The plume folds into darkness. The red plumes of the tube worms fade first, then the white tubes, then the rock itself, and the vent continues in the black water without needing to be seen.